Okay, good morning, everyone. Welcome to today's series of lectures, which are part of the EMBO Lecture Tour. We have three outstanding speakers today, and as we go through the day, but let me begin by introducing the first speaker, Professor Michael Hall. So Mike is a truly international scientist. He was born in Puerto Rico and lived parts of his early life in Venezuela and Peru before he moved to the United States to do his education in college and subsequently PhD, for which he studied at Harvard, looking at the outer membrane proteins, the porins in E. coli. And after that, he, after postdoctoral training, also in the US, he moved to the Biozentrum in Basel, where I think he's been ever since. Mike has made many important discoveries. They all stem from his, at the time, an observation that identified two proteins that mediated the effect of rapamycin on growth. And these turned out to be what he called TOR1 and TOR2. And subsequently, when mammalian world woke up to this and discovered the orthologs, they added the word M, and now everybody knows it as mTOR. Mike has, of course, been honored a lot, and I would eat into his speaking time if I were to go through the list. But I'll just mention that he's been awarded the Breakthrough Prize and also the Albert Lasker Prize for Medical Research. He's a member of EMBO and also a member of the National Academy of Sciences. But perhaps there can be no greater honor to a scientist than to make a discovery and find many years later that it has influenced our thinking of how cellular biochemistry is and metabolism are regulated, and that it has broad implications well beyond the original system in which one found it. So without further ado, Mike, we look forward to your talk.
Good morning. Thank you, Raghu, for that very nice and insightful introduction. I have to say, it's also a huge pleasure to be here. This is a wonderful place. Beautiful, thoughtful. I suppose you all know you're very fortunate to be here. So I don't have to tell you that, but it's a real pleasure to be here. It's certainly one of the leading institutes in the world, and I'm very glad I finally had the opportunity to visit. I should say some words about EMBO too, since we're being sponsored in part by EMBO. EMBO stands for European Molecular Biology Organization. I'm sure many of you know that. This is an organization which is a very helpful organization, and there's a special relationship between EMBO and India. So those researchers in India have many advantages within EMBO. For example, Indians can be Young Investigator Program fellows. They can be EMBO Global Research fellows. There are many opportunities, and it's not just for Europeans, but also for Indians. So I encourage you, if you don't know about EMBO, to look it up. You might find something that could, particularly for the young, but also for the not so young, could help your careers. Okay, we work on cell growth control. We work on TOR signaling in particular. And what I'd like to do today is tell you two stories, if I have sufficient time. And both of them deal with our long-standing efforts to understand the role of TOR in disease. As you heard from Raghu, we can use TOR or mTOR interchangeably. So don't be confused if I don't say mTOR. These two stories I'm going to tell you deal with two different diseases. One is cancer and the other is diabetes. So the main story I'll tell you is the cancer story. And at the end, if I have time, I'll tell you the diabetes stories. The cancer story is the major story. It's the larger story I'm going to tell you, the more advanced story. But I'm more excited about the diabetes story at the moment. So I couldn't decide which one to tell you. So I'll tell you both. And I'll try to get through them both. If I go a little over time, please forgive me. But before I do that, I'd like to give you an introduction to TOR signaling. This is a very large, complex field now. And it seems it's very easy to be overwhelmed for those outside the field trying to penetrate into this field. So, and that's largely illustrated in this first slide. So this here is the very first ever model of Tor signaling published. It's one we published shortly after our discovery of Tor in 1991. Now, I have to say I'm kind of ashamed I published this model because there's almost nothing of value in it other than to say Tor exists. It's hardly a model. It's more, who here knows who Gary Larson is? Cartoonist, science cartoonist? Yes, this looks more like a Gary Larson cartoon than something you'd find in a respectable scientific journal, although we published it in a respectable scientific journal. There are more question marks here than there's actual valuable information. Today, the picture is considerably more complex, and this is already woefully out of date. This is an old slide. And this increase in complexity, a rather rapid increase in complexity, has been driven by the fact that Tor has turned out to be both fundamentally important and clinically important. So there have been two very large and research communities which have been investigating this, which has led to this exponential increase in publications in this area. So let me briefly take you back to an era closer to this, and by the end of the introduction, I hope to have you something, have your level of knowledge something closer to this. And I'll do this in a very abbreviated way, so buckle your seatbelts and let's go. The story actually started earlier than 1991. It started in 1965 when a group of scientists went to Easter Island in the South Pacific to collect a soil sample. And what they were doing is they were prospecting for exotic microbes, which they hoped would produce some novel metabolite, which they could then develop into a drug. And what they were looking for in particular were antifungals, a new antifungal. They did isolate such a bacterium from the soil sample. They did isolate such a molecule, secondary metabolite from that bacterium, and they called it rapamycin in honor of the name the locals used for the island of Easter Island, which is Rapa Nui. So it's named after Rapa Nui. And as they were developing this drug this molecule as a antifungal, they discovered that it had the undesirable side effect of suppressing the immune system. Of course, this is the last thing you want to do to someone with a fungal infection. So it was very quickly dropped and rejected and not further developed as an antifungal. But about 10 years later, a group of very bold transplantation surgeons were developing transplantation surgery as a viable clinical procedure. And a few molecules played an instrumental role in this development in medical history, and rapamycin was one of those. So they applied now rapamycin as immunosuppressive to be able to do transplantation surgery. Other drugs, for example, are cyclosporin A and FK506, are also well-known immunosuppressive drugs. So it's now rediscovered, but repurposed, but now for the purpose or the use or for the effect which had originally been rejected, to suppress the immune system. And it became very famous as an immunosuppressive drug. If you had an organ transplant in 1980, likelihood of survival was very low. If you have an organ transplant today, the likelihood of survival is very high due to these drugs such as rapamycin. If you go to Rapa Nui or Easter Island, you'll actually find a plaque where to commemorate this act of picking up this handful of dirt, which led to this medical revolution. And this is a picture of that plaque. And this says, it's in Portuguese, in this place was obtained in January of 1965, samples that not only allowed the isolation of rapamycin, but also ushered in a new era in transplantation surgery. Now, having worked on rapamycin most of my adult life, I've always wanted to go to this shrine and pay homage. I finally managed to do that a few years ago. We flew all the way to Rapa Nui, which is about as far away from Switzerland as you can possibly get. It's the most isolated spot in the world. We dropped our bags off at the hotel and immediately headed out to see this place, this plaque. We had to stomp through the brush to get there. And what we found was, so this is our little party of scientists. What we found was the plaque had been stolen. So I still have not seen the plaque. If someone knows where it is, let me know and I'll try to like to see it. Okay, back to more serious matters. So this is rapamycin, this natural molecule secreted by the soil bacterium. It now has applications in 3 therapeutic areas. It was first became famous as an immunosuppressive, but now it's actually even more famous as an anti-cancer drug. And it's now also used in cardiovascular disease, in particular treatment of restenosis after angioplasty. So this one molecule that had very modest beginnings in the dirt in this faraway place has applications in 3 major, very different therapeutic areas. So this should already tell you or give you a hint that there's something unusual or special about the target of this drug. How can this drug targeting one thing be used in so many different therapeutic areas? So this is, and I'll get back to that later. So what is TOR? TOR, or what is the target of rapamycin? The target of rapamycin, the protein we discovered, the protein that rapamycin binds directly, we call this, we discovered we called it TOR, after a very imaginatively targeted rapamycin. And we did this in yeast. And there were 2 TOR genes in yeast, and therefore 2 TOR proteins in yeast. And they turned out to be the founding members of this novel class of atypical protein kinases, serine-threonine kinases. And what this class of kinases have in common is that they all look like lipid kinases, PI4 and PI3 kinases in particular, but all are protein kinases, and none has been shown to actually be a lipid kinase. And to this day, we don't know the significance of this resemblance to lipid kinases. Since our earlier discovery of TOR in yeast, it's now been found in all eukaryotes, including worms, flies, plants, and mammals. Usually there is the TOR, we originally gave it from yeast, but with a prefix to indicate what organism it comes from. mTOR used to be mammalian TOR. Now this has been mutated to mechanistic TOR for reasons I can talk to later, which I'm not so happy about. But mechanistic TOR is now TOR from all organisms because it doesn't specify which species. And of course, this creates confusion because in the literature, mTOR was used as mammalian TOR in the past, but just so you're aware of that. Now, yeast was unique in that it had these 2 TOR genes, but all other eukaryotes have a single TOR gene and a single TOR protein. So for once, the simple eukaryote was more complex than higher eukaryotes. But this actually helped our analysis for reasons I might get to later. So what is the function of this very highly conserved kinase? Well, its role, as we discovered through a long process of a rather long, confusing process with many misleading pathways, was to control cell growth and thereby cell size. And this is most, I think, photogenically illustrated in experiments done in Drosophila. So these are experiments done by Tom Neufeld and Bruce Edgar, both phylogeneticists. In this case, Tom Neufeld selectively inactivated TOR in these 2 GFP expressing cells. And as you can see, these cells are smaller than all the surrounding wild type cells. And this is the fat body of the fruit fly. Bruce Edgar did the inverse experiment. He hyperactivated TOR. In this case, he sees the cells are larger than all the surrounding wild type cells. So the obvious conclusion is that the role of TOR is to control cell growth and thereby cell size. Now, Tom Neufeld took this experiment one step further. He isolated a mutant fly in which all cells of the entire fly are defective for TOR signaling. And this is the what he obtained. This is a so-called hypomorph. It's a partial loss of function. If it had been a complete loss of function, this fly never would have been born because TOR is absolutely essential for viability. But as you can see, this fly is smaller but perfectly proportioned. And then Tom Neufeld asked the question, why is this fly smaller? Is it smaller because it has smaller cells, but the normal number of cells, or because it has fewer cells, each of which might be normal in size? So is it a matter of cell size or cell number? And he could address this question by looking in the wing, where you can very easily measure the circumference of cells and also count the number of cells, because the wing is a plane of cells. And what he found was that this fly has more or less the normal number of cells. And of course, from this, you extrapolate to the rest of the animal. And he realized that this fly has more or less a normal number of cells, but each individual cell is slightly smaller to give rise to the smaller fly. So the obvious conclusion here is that TOR controls not only cell size, but thereby also controls sizes of tissues and ultimately size of the whole animal. More recently, TOR has also been shown to control growth in many other physiological contexts, often independent of cell division, as would have been the case in this developing fly. In other words, in post-mitotic contexts. Here are some of my favorite examples. In the liver, if you fast or feed, your liver changes in size, and this is due to your existing liver cells increasing or decreasing in size. You don't acquire more cells or lose cells. As you all know, if you exercise, your muscles get bigger, and we know this is due to, again, increase in individual muscle cell size, not an increase in cell number. This is also, like liver, has been shown to be TOR controlled. In the central nervous system, we have very dramatic changes not only in cell size, but cell shape. This has also been shown to be TOR controlled. And someone has actually shown that queen bees are bigger because TOR is upregulated in the larvae, which give rise to these queen bees. So the take-home lesson here is that TOR controls growth very broadly, broadly in terms of physiological context and broadly in terms of organisms. Essentially, any case in eukaryotic biology where you see a change in cell size, regardless of physiological context, there's a very, very high likelihood that TOR signaling is somehow involved. So the next question was, how does TOR control cell growth, cell size? Well, again, here, this is a process which took us and others many years, but we found that TOR controls a very large number of cellular processes which collectively determine mass accumulation. They're all involved in, well, they all control mass accumulation and therefore cell size. And these cellular processes can be divided into two subgroups: the anabolic processes, which TOR activates, and the catabolic processes, which TOR inhibits. So what TOR does is it balances these opposing forces of synthesis and degradation, such that the right level of mass accumulation occurs to elicit the appropriate cell size relative to whatever the available nutrients are, the quality and quantity of nutrients. So in very simple terms, TOR is what makes us grow when we eat, grow in a controlled manner. So this has led to the notion that TOR is a central controller of cell growth, just like cyclin-dependent kinases are central controllers of cell division. So how does TOR actually control all these processes? Well, there are effector signaling pathways which emanate from this highly conserved kinase to intersect key proteins involved in all these processes to control their activity. Here's a simplified overview of the TOR signaling pathway. Oh, let me here, yeah. This is a picture of the mammalian TOR signaling network, and I call it a network, not a pathway, because it's in fact more than a single pathway. It's at least two pathways, and the network is defined by these two complexes, which we discovered in 2002, which we call TOR complex 1 and TOR complex 2. So each of these two complexes are not only structurally but also functionally distinct. They each phosphorylate their own substrates to control different cellular processes. So TOR is in fact two distinct kinases, depending on which complex it is part of. Now, like TOR itself, these two complexes are also conserved all the way from yeast to mammal. So the picture that has emerged is that this is a fundamental or primordial ancestral signaling network, which has been conserved throughout eukaryotic evolution to control this very fundamental process of cell growth. Now, that statement I just made is not absolutely true, because this part of the network here evolved much later. It wasn't there already from the origins of eukaryotic cell biology. And this is the growth factor or insulin signaling pathway. This evolved with multicellularity. Didn't exist in unicellular organisms. The reason for that is that the reason for that is that cell growth control in multicellular organisms, in metazoans, is more complex than growth control in unicellular organisms, because in metazoans, it's critical to control the growth of every cell relative to every other cell such that our organs end up being properly proportioned. So you need some growth control over a whole body plan. And that then is what the growth factor input provides. It controls growth over a whole body plan. And once you have this input controlling growth over a whole body plan, along with local nutrients, this is the original input already existing in unicellular organisms controlling TOR. So local nutrients and local energy in the form of ATP, the whole network is activated, anabolic processes are turned on, catabolic processes are turned off. There's macromolecular synthesis, mass accumulates, and the cell grows in size. Another fascinating aspect of the TOR signaling network is the unusually large number of diseases it's been implicated in. And this have been, well, the number of diseases actually been functionally linked to. And this has been discovered as genes in causing all these diseases have been identified and shown to encode components which are members of the components of the mTOR signaling network. This includes, and what all these diseases have in common is they're all characterized by inappropriate or ectopic cell growth. Very consistent with the notion that TOR is a central controller of cell growth. And this can be malignant forms of cell growth or benign forms of cell growth, but in all cases, inappropriate cell growth. More recently, TOR has been implicated in another set of disorders, the so-called metabolic disorders. We know chronically high circulating levels of nutrients can upregulate TOR even in the absence of growth factor. This can lead to adipogenesis, obesity, and type 2 diabetes. But there's a more direct link here in the network where TOR phosphorylates IRS in the insulin signaling pathway to inhibit it. This then leads to loss of insulin signaling or leads to insulin insensitivity, which, as you know, is a hallmark of type 2 diabetes. In this context, and this then provides a link between diet and diabetes. In this context, some have actually proposed rapamycin for a fourth therapeutic area, diabetes, because if you inhibit TOR with rapamycin, you would inhibit the negative feedback loop and you would restore signaling through the insulin pathway. Nobody, to my knowledge, has actually developed rapamycin as an anti-diabetic because there are a number of complications which would you would think would preclude that. But something which gives credence to the notion that it might be possible is that the world's most commonly prescribed anti-diabetic, metformin, acts at least in part by inhibiting TOR. In this case, via activation of AMPK, which is a negative regulator of mTOR signaling. So I've given you a very quick overview of our efforts over the last 30 years to understand the function and physiology of TOR and TOR signaling. In this time, we've also been trying to understand TOR signaling at the atomic level. So we've also determined the structure of TOR complexes. Here is mammalian TOR complex 1. It's a very large complex. It's almost the size of a ribosome. And it's what we learned is it's a dimer of heterotrimers. So there are two TORs here. Here's one TOR and another TOR. These are the Raptor subunit and the List H subunit. And because it's a dimer, it has two catalytic sites. The catalytic site is in this cleft here and here. TOR itself has a rather standard structure for a protein kinase, a bilobe structure with a catalytic site between the two lobes. I think the most interesting thing we learned from the structure of TOR complex 1 was the mechanism of action of rapamycin. How does rapamycin inhibit TOR? So well, what I've not told you yet is that rapamycin alone cannot inhibit TOR. It first has to bind another protein in the cell called FKBP, and this FKBP rapamycin complex then can inhibit TOR. So rapamycin hijacks another protein to be able to inhibit TOR. Rapamycin alone can bind TOR, but cannot inhibit it. So we never understood why that was the case. Why can rapamycin alone, like most drugs, binding something alone, not inhibit TOR? Why did it first have to bind this other protein called FKBP? And the answer came from the structure. So keep your eye focused here and here. The FKBP protein will appear. So this is FKBP. And what rapamycin does is it glues FKBP to the lip of this catalytic cleft, leading to the notion that rapamycin is a so-called molecular glue. This explains the mechanism of action of rapamycin and why FKBP is absolutely essential for rapamycin inhibition. And that is because the FKBP forms a lid over the catalytic cleft, which blocks access of substrates to the catalytic site down here. Rapamycin alone cannot inhibit because it simply does not have enough bulk or mass to block access of substrates to the catalytic pocket. But this molecular glue concept is now becoming very, very popular in cancer research today in many other contexts. And I'll come back to that later. But you can think of rapamycin as the first molecular glue. Okay, we also determined the structure of Tor complex 2. Even more recently, this was down to 3.2 angstroms. And I think the most interesting thing we learned, well, it turns out is also a dimer. It's a dimer of hetero tetramers, so there are more subunits to the Tor2 complex. And I think the most interesting thing we learned from the structure of Tor complex 2 is something that had puzzled us since 2004, where we found out that Tor complex 2 cannot be inhibited by rapamycin. We never understood why it was the case. Why can rapamycin FKBP bind Tor and TORC1, but can't bind Tor and TORC2? And the structure revealed the answer. And the answer is that this subunit, which is specific to Tor complex 2, masks the FKBP rapamycin binding site. So the rapamycin FKBP simply cannot bind to Tor complex 2. Okay, so that's my introduction. Let me now get into the main story. And this involves cancer and cancer metabolism. Back to the Tor signaling network. So this, it's been known for many, many years that Tor can drive cancer. Not surprising. It's a controller of cell growth, and cancer is a growth-related disease. So we wondered, but it was not known how Tor promotes cancer. So this is something we want to address. So to do this, we made an mTOR-driven mouse model of cancer, in this case, liver cancer, hepatocellular carcinoma. We did this by deleting two tumor suppressors upstream of the Tor complexes, TSC1 and P10. And we deleted these at the same time, specifically in liver cells. So if we delete TSC, this hyperactivates TORC1. If we combine that with a deletion of P10, this further upregulates TORC1, but now also upregulates TORC2. So what happens when we hyperactivate TORC1, TORC2, specifically in the liver? So this is what we see. So these are control livers from our so-called liver double knockout, liver-specific double knockout mice. And we find the first thing we see is the enlarged liver, or hepatomegaly, and the liver continues to grow in size due to the growth-promoting effect of hyperactive Tor in the hepatocytes of this liver. And this liver continues to grow for the lifetime of this mouse, so it becomes a large part of the total mass of the liver of the mouse. We then see non-alcoholic fatty liver disease appearing at about 6 to 8 weeks. This then progresses to an inflamed liver, which eventually, by about 16 weeks of age, manifests as very mild HCC. And then by 20 weeks of age, there's very severe HCC, and these mice have to be sacrificed at this point. So we want to understand what is giving rise to these tumors in the liver in this Tor-dependent manner. So to address this question, we excised individual tumors and applied a whole slew of omics analyses to them. And I'd like to start by focusing on the metabolome. So we isolated these tumors, we extracted metabolites, we did mass spec, we identified about 16,000 metabolites, of which about 4,000 were annotated, and of these 4,000, about 1,000 either went up or down in the tumor relative to wild-type tissue. And then we did pathway enrichment analysis of these 4,000 changed. The pathway which was most affected was amino acid metabolism. So we then did target analysis, looking, assaying the levels of all the individual amino acids. And we find that in some cases, amino acids were down, but the most significant effect, and one uniquely where the amino acid concentration was higher, was arginine. So we immediately wondered why is arginine high in the tumor. So we looked at the pathways which synthesize arginine. Arginine is made as part of the urea cycle, whose role is to detoxify ammonium. It converts ammonium to urea, and then urea is secreted from the cell. But what we found, much to our surprise, despite the very high levels, concentrations of arginine in the tumor cells, all the enzymes which make arginine, these urea cycle enzymes, are turned off. And let me introduce you to this key, because I'll use it in other slides. The box on the left here represents mRNA, and the box on the right is protein. And this data comes from our transcriptome and proteomic analysis from these same tissues. So you can see all these enzymes are shut off at the transcriptional level. We were then left to wonder why are arginine levels high if the synthesis is down. And the first thing, of course, that comes to mind is that arginine import must be increased. So we looked at, well, there are several arginine importers, and they fall into two classes: the uniporters, which bring arginine in a facilitated manner, or antiporters, which require an anti-solute to be secreted to import an arginine. So it exchanges one solute for the other. And for it was originally thought that glutamine was the anti-solute, which is exported to bring in one arginine. And there are several in each class. So we looked at all these proteins, and we found of the 7, most of them indeed were up. We could detect them at the transcriptional level here. We couldn't detect these proteins by mass spec because they're membrane proteins, and those are often difficult to detect. But we can confirm here that they are indeed up at the protein level. In the meantime, the Christoph lab has shown that it's not glutamine that's important as anti-solute, but asparagine. And indeed, we also saw that asparagine synthetase is increased in the tumors. In other words, everything you need to import more arginine is upregulated in the tumors. So we actually, of course, did an arginine transport assay and found that arginine import is increased in these tumor cells. So this explains the mystery, at least part of the mystery, of why there's high arginine concentration in these tumors, but doesn't answer the question: is arginine important for tumorigenicity? Is this just some random consequence of something that's immaterial? So we went back to our mouse model and put them on, but with mice put on different diets: normal arginine levels, 10% of arginine levels, and 1% of arginine levels. And even at 1% of arginine levels, there's sufficient arginine for normal growth. We still see the hepatomegaly. The mice are fine. There's vast excess of arginine in the diet of the mice. But we saw a dramatic decrease in the number of tumors when we decrease arginine. So the arginine indeed seems to be essential for tumor growth. And if we looked in the cells, assayed arginine levels, in the non-tumor tissues of these livers, we saw arginine levels go down as expected. But in the very few tumors that still appear, even at 1% arginine, there was still high arginine in those few tumors. Again, a very strict correlation between high arginine and tumorigenicity. So again, telling us that the arginine is important. So what's it important for? Well, arginine is a very versatile amino acid. It's used for many different things: protein synthesis, used to synthesize other amino acids. It's one of the important signaling molecules for to activate TOR. It's arginine is one of the key nutrients which activates TOR signaling. It's also a precursor for creatinine, nitric oxide, and polyamines. We could root out all these, and we started focusing on polyamines. So this is how polyamines are made from arginine. So again, here we are now, the full urea cycle. Arginine is made here. It is converted to polyamines. So these are the polyamines. These are small cationic molecules, essential for cell viability, but nobody really knows their molecular function. It is said they mediate cellular homeostasis, which is a nice way of saying we have no idea what these things do. And the famous polyamines are putrescine, spermidine, and spermine. And they come from arginine via 2 parallel pathways. One is the key enzyme agmatinase, and the other one is the key enzyme arginase 1. So they can go either this way or this way to produce putrescine, and then putrescine is used to produce the other polyamines. But what we found was that these two, the key enzymes involved in conversion of arginine to polyamines, are also decreased, and that is shown by many different approaches here. But despite the reduced polyamine synthesis, like arginine synthesis being decreased, polyamine levels are also high. And this is again due to increased polyamine uptake. But the important point is that the arginine pool in the cell is not used to make polyamines in this case, because the polyamine, which is also high, comes from the tumor microenvironment, like the arginine itself. So the cells do not accumulate arginine to make polyamine. So we thought that the role of, so why is polyamine synthesis down? Well, we thought it's probably down to maintain the high arginine levels. And we could do the experiment going back to our mice. We can restore expression of arginase or AGMAT, so that arginine is now again metabolized to make polyamines, and see what effect this has on tumors. And indeed, if we consume the arginine in these tumor cells, we lose tumorigenicity, and number of tumors goes down. Okay, and again, arginine levels go down in the tumor because it's being consumed now to convert to polyamines. So this adds also to the answer of how arginine levels stay high. It's not only an increase in uptake, but it's also a decrease in consumption. But this also tells us that what's important is not arginine being metabolized to something else, it's unmetabolized arginine, which is required for the tumorigenicity. So arginine itself. So what could this arginine be used for to drive tumorigenicity? And here in the literature, we saw a nice example which gave us some inspiration. In the case of T cells, it's known that arginine is required for T cell activation, and it does so by enhancing oxidase phosphorylation and nucleotide synthesis. So there's an effect of arginine on metabolism in T cells. So we wondered whether this would also be the case in our liver tumor cells. So we sequenced we did determine sequenced the mRNA in tumor cells with high or low arginine, and we found that indeed many metabolic pathways were changed at the transcriptional level. And from this, we generated a gene signature, which I'll come back to, which represent the different metabolic pathways which change upon high arginine. And this includes NAD, amino acid, pyruvate, aldehyde, glycolysis pathways. So this is where we were at this point. This is a normal cell. Arginine synthesis is being driven by the urea cycle, and this is used to make polyamines. But in the tumor cells, we lose all these enzymes. The cells compensate by increasing arginine and polyamine uptake. And this then arginine leads to some metabolic reprogramming at the transcriptional level, including asparagine synthetase increase in expression, which then makes asparagine, which brings in more arginine. So this functions as a positive feedback in this mechanism. Since this is at the transcriptional level, we hypothesize that there must be some arginine binding transcription factor in between. That's how arginine is doing this. So we set out to discover what this protein might be. And to do that, we made an arginine column. So fixed the arginine to a matrix, ran extracts over it from our tumors, from the liver tumors in our mice. And we took our 42 top hits, or proteins which bound to this column, knocked them all down, and looked for one, and then assayed for asparagine synthase expression. We found indeed one protein, knocking down one protein resulted in reduced expression of asparagine synthetase. And this protein, RBM39, is an RNA binding protein. It's known to be a pre-mRNA splicing factor, but it's also a cofactor in transcription. It determines transcription. It can be a transcription coactivator or co-inhibitor. So that we thought this would be how arginine could be controlling metabolism or transcription of metabolic genes. We confirm that arginine binds its protein very specifically in the N-terminus region, where there's an intrinsically disordered region. So we think arginine perhaps binds it, induces order, and activates this protein some way. But that's ongoing work. But to confirm that indeed is a factor, if we knock down RBM39, we can reverse all the effects of high arginine. So high arginine is indeed binding this protein to control transcription of all these metabolic genes. Interestingly, we could also confirm this in another way, and this I'm back to molecular glues. Molecular glues are causing lots of excitement in the cancer area because there are many small companies starting up now to identify a molecular glue which will target your protein of choice to a ubiquitin ligase. And this will then lead to ubiquitination of that protein, its degradation and elimination. And there happened to be a glue, just by chance, a molecular glue called inducilam, which targets RBM39 to ubiquitin ligase in a way which RBM39 never encounters this ubiquitin ligase, but it leads to its ubiquitination and degradation. So it's a good way to knock down RBM39. We could do this. We see again when we treat cells with inducilam, we reverse all the effects of arginine. We could also give inducilam to the mice, and this also blocks formation of tumors. So we now not only have a mechanism, there's also a molecule out there to target this process. So this is now the final model. We have RBM39, which binds arginine, which then leads to metabolic reprogramming by turning up or turning down transcription of these target genes as this cofactor. Okay, I just have some minutes left. I'd like to tell you the second story, which is one I'm really excited about. And this is who here knows that there are sensory neurons in fat, adipose tissue? Okay. How many neurobiologists are there here? No. Oh. Okay. Well, I didn't know either, I have to say. It was a big surprise to me. But that's one of the discoveries we made here. And this story goes back 15 years when we knocked out TOR complex 2, specifically in adipose tissue. And what we found was that knocking out TOR specifically in adipose tissue had systemic effects. So they're non-autonomous functions of TOR, of adipose TOR. And these systemic or body-wide effects included hyperinsulinemia and insulin insensitivity. So we reasoned that there must be a mechanism where there's a TORC2 dependent factor secreted from adipose tissue, which acts on liver and particularly beta cells to reduce insulin synthesis. And the logic of this would be that TORC2 itself is controlled by IGF and insulin as part of this growth factor input. So high levels of insulin would, it's a way of maintaining insulin homeostasis. If insulin gets too high, TORC2 is activated and it shuts down the insulin production. In the absence of this, these both accumulate to high levels and you get this systemic effects. So we always hypothesize there must be a secreted factor, something secreted in the bloodstream, which goes and acts on these other tissues to control expression of the genes encoding these proteins. We spent 10 years looking for this factor, always failing. It was sort of the rite of passage for each new postdoc in the lab to go after that factor, to go on the hunt for that factor. Everyone always failed. So a student came in the lab and wanted to take another approach. She wanted to go back to the beginning and repeat this experiment, but not use this driver, this Cree driver to delete TOR, which deletes at birth before adipose tissue is fully developed. And I said, okay, because we're also worried that this might be a developmental defect, not a specific defect to TORC2 in adipose tissue. Because if the knockout occurs before the tissue is fully developed, there could be many indirect consequences. So she went back now using another Cree driver, but this is a tamoxifen inducible Cree driver, so you can knock out in the adult after the fat tissue is fully developed. She did this, but she tamoxifen treated for 5 days and then took different time points after deletion to study the mice. this is what she found. She found that these mice were hyperinsulinemic, just like we'd found many years before. So it was not a developmental defect. And she also found they become insulin insensitive. This is an insulin tolerance test to measure this. But this is a slow response. This occurred only at 2 weeks or 1 to 2 weeks of age, and then 4 weeks or 1 to 2 weeks after knockout of TORC. At 3 days after knockout, the mice were not insulin insensitive, but the hyperinsulinemia was right away. So she was wondering what gave rise to these effects. So she went back now to mass spec. She isolated fat tissue from our knockout mouse and from wild type mice, did mass spec, did pathway enrichment again, and found that much to our surprise, what was changed in expression were neuronal proteins, synapse proteins in particular. And this, of course, threw us for a loop because we thought we were studying adipose tissue, not neurons. But it turns out the well-characterized sympathetic nervous system, which innervates the adipose tissue. So she looked at the sympathetic nervous system in fat. She had to develop a whole mount 3D imaging method to do this, but this is what she found. So this is a sympathetic nervous system in a fat pad of the mouse. The marker is tyrosine hydroxylase. You see this very beautiful network of nerves in the tissue. Of course, she then looked in the knockout, but she saw absolutely no difference between the control and the knockout mouse. No effect on the sympathetic nervous system. In the meantime, we also learned that there were very few reports in literature, very old reports, saying there might also be a sensory nervous system in adipose tissue. And this is the work of essentially one man by the name of Tim Bartness, who died many years ago. But in the techniques of his day, he had some indication there were sensory nerves in adipose tissue. So we went back and obtained a marker to look at sensory neurons, and much to our delight, and we indeed confirmed what Bartness proposed. There was a network of sensory neurons in adipose tissue, not as dense or developed as a sympathetic nervous system, but a network nonetheless. Irina then went back to the mutant mouse, and she found this network disappears in the knockout. So these are nerve bundles coming in from the dorsal root ganglion into the fat pad, but then they don't. There's no arborization and innervation of the parenchyma of the fat. And that's again illustrated here. This is control TORC2, in which she's co-stained now in the same fat. You can see the two separate nerve networks in the adipose tissue, and in the knockout you see only one. Now she wondered why was TOR required for this sensory nervous system in fat, and to do this she went back now to her mice at different time points after deletion of TORC2. So here's after 3 days of deletion, the network was completely normal. At 2 weeks, it's already largely gone. We have a time point of 1 week where it's somewhere in between these two, and again by 4 weeks it's completely gone. And this is all quantified down here. So what happens is a TOR complex 2 in adipose tissue is required to maintain nervous neurons in the tissue, sensory neurons, not required for establishment of that nervous system. So what is it doing? What is sensory system sensing? Well, we don't really know, but what might its function be? And here then we can go back to the phenotypes that we see upon loss of TORC2, and the phenotype that it correlates with extremely well, even other time points we've looked at, is disappearance of the nervous system. As the nervous system disappears, we start seeing appearance of the systemic insensitivity. There was a beautiful correlation here. So this has us very excited because it tells us that perhaps fat is not signaling to the liver and beta cells by secreting some factor, which we never found, but indeed signaling via this heretofore unknown sensory nervous system to the brain, and then the brain is signaling perhaps also through nerves to these other tissues. This is a bit of a breakthrough because it gives us a completely new perspective on how to think of diabetes. It's not just factors signaling, but in this case, it's also the central nervous system is how the body responds to fat to lead to diabetes. So these are my conclusions from the second part. There is in adipose tissue a sensory nervous system. I would say we confirm this original work of Tim Bartness. TORC2 is essential for this sensory innervation. Adipose tissue thus might be mediating these whole body effects, this energy homeostasis or glucose homeostasis via the central nervous system and not through secreted factors as we thought. And finally, we're also excited at the possibility that this might be an explanation for something which for which there's no explanation to date, is this diabetes-induced neuropathy. And this is a disease, well, as a consequence of diabetes. Diabetics often develop numbness in their fingers and their toes, often that, and this is due to death of neurons in these in the neurons and toes, and this leads to lesions and sometimes loss of toes and fingers. Nobody understands why insulin leads to this effect, but we now believe, or are testing now, it's actually perhaps loss of insulin signaling in the fat in the under the skin in the fingers or the toes, which is causing these nerves to retract, and then leads to this numbness and loss of sensation in the periphery. Is of course something we can test very easily, and we're going to do that. And having this information, no one had ever made a link between fat and this phenotype, or even TOR in this phenotype. So now we have something very direct to test, and of course this could lead to a very interesting treatment for this side effect of diabetes. Okay, I'm sorry I went over time. Here are the people who I give credit to for this work. The first project was largely the work of a very good postdoc, German postdoc by the name of Dirk Mossman. The second part on the sensory nervous system in adipose tissue, a student by the name of Irina Frey. And these are some of our collaborators with different aspects of what I told you today. Okay, thank you for your attention.
Thank you, Mike. Questions? Stock. I would like your perspective on the role of mTOR in aging. I mean, I've often had it a bit confused in that we know there is a loss of protein synthesis and there's loss of muscle mass and so on during aging, but yet we find that the inhibition of mTOR much later in life, inhibiting this protein actually seems to restore some of this. What's your thoughts on how this works?
Oh, I can go lecture for an hour on that. I don't think you want that. It seems like what we do know is that all the processes via which TOR controls growth, activates growth, for example, are the same processes via which it shortens lifespan. Okay, so you need too much protein synthesis leads to shorter lifespan. So mutations in protein synthesis, protein biosynthetic pathways have led to longer lifespan. Proteins, autophagy has been shown to control lifespan. TOR controls lifespan, the appropriate way for that. On and on, all the things we've seen that TOR controls also control lifespan, and if TOR controls them up, they're being high is what shortens lifespan. So everything fits perfectly. And what's behind this is this leads to what I think is called the antagonistic pleiotropy model of aging. I don't know if you're familiar with this model, but the idea is that during what evolution selects, there's selective pressure for growth. This is, selective pressure optimizes growth until the point we or whatever reaches reproductive age and pass on our genes. And once that happens, there's no selective pressure. We are sent adrift. We've done our evolutionary purpose and fulfilled our evolutionary purpose, and selection has no interest in us anymore. So everything that is optimized for growth should be turned off after reproductive age, right? But it's not turned off. There's been no selection for a mechanism which would turn off TOR signaling, and it remains at this high unabated level. And therefore, with rapamycin, we can reduce it artificially and extend lifespan. That's the theory.
Fascinating second part of the talk, the diabetes story. So I'm just trying to imagine how the reduction of TOR activity or the loss of TOR activity is making those neurons disappear. I mean, how do you relate it back? Is it actively triggering the death of those neurons through a new mechanism to be discovered? Or are you imagining it related to the lack of cell growth? I mean, relating to TOR's activities.
We don't think it's cell growth. It's not here in this case. It's TOR controls the growth of the cell in which it resides. I think the effect on the neurons is a separate process, because this is not the TOR is controlling neurons which are other cells. I don't think it's apoptosis, because the main bundles of neurons are still there, and it's one set neuron that goes all the way back to the dorsal root ganglion. And if that cell became apoptotic, everything would disappear, not just the arborizations. So I don't think that's the case. What I think is going on is that, and this is my favorite theory, and we're testing, is that TORC2, which as part of its in cell growth control, controls lipogenesis. It activates fatty acid synthesis and lipogenesis. What I think it's doing in that process, it's also making some bioactive fatty acid, which is probably secreted, which is acting on neurons, which is somehow stabilizing them. We're trying to identify what that might be at the moment.
That was a very nice talk.
Yeah.
My question is more with the first part of the talk. You spoke about RBM39 and validation of that target, right, in the entire pathway, mTOR signaling. So you have compared knockout versus molecular glue, right, in terms of validating the target. So my understanding of molecular glue is it tends to be very sticky and brings in a lot of proteins within a signaling pathway, causing degradation. So how does your omics data compare if you look at knockout of this target, your RBM39 versus molecular glue mediated degradation?
Well, these molecular glues are not non-specific. They're extremely specific. That's why they're valuable as drugs. The term sticky or glue is probably not the best term, but like rapamycin is a molecular glue, and it is probably the most specific drug in the world. It binds FKBP and TOR and nothing else. So these, first of all, they're not generally sticky molecules. They're very specific. They're adapters between the two. They're very specific contacts which are created to be. We have done transcriptomics upon, I'm trying to remember, we did RBM knockdown or Indisulam treatment, but you want us to compare those two. I know we've done one of those two, but not both. So I can't do the comparison. But what came out of that was, since RBM39 has many roles in transcription and pre-mRNA splicing, that analysis told us that the arginine-regulated activity of RBM39 is its role in transcription activation or inhibition, not on pre-mRNA splicing.
Thank you.
Hi, yeah, it's a nice talk. I have a quick question. You said that in cardiac diseases, cardiac hypertrophy, right, where actually it is not a cell increase in cells, whereas it's increase in size.
Yeah.
Rather in cancer, it's basically opposite.
Yeah.
But yet you term both as growth. Can you comment on this?
Well, you need to get proliferation. You need a process of growth and division. Okay. In the case of hypertrophy, you don't get, the division doesn't occur, so the cardiac muscles just get bigger and bigger and bigger. In the case of cancer, you have cell division too, cell numbers increasing. You have to regulate both the growth part and the division part. And that's why you get more cells, you get a tumor. In some cancers, the cells are even bigger in addition to being more in number because the growth part outstrips the division part.
But the signaling cascade, you said it's same, but it's opposite, right? Because here you see a cell size increase, whether the cells are not dividing.
Yeah.
Whereas in the cancer, the cells are dividing. So
Right.
obviously there is an opposite cell signaling happening here, right?
No, I don't think you need an opposite. If a cell, two things can control cell size. One is just increase of mass
Protein cells, yes.
and no division, or the time of which division occurs. If division occurs early, even if there's an increase in mass accumulation, the cell will be smaller.
Okay.
Right? It's just when you
Just
decide
the
to
term,
divide.
yeah.
The key question is, but that's not usually the case. In normal cells, they know to reach a certain size and then divide. There's still an open question how they know they reach that size. That's still ongoing work.
Hey, Mike,
Yeah.
very nice talk. This first part, the TACP10 double knockout, what makes it HCC specific?
The double knockout in the liver.
Yeah.
Well, we use the albumin Cree to just it's a liver specific Cree driver.
Okay.
Yeah.
So related on that same part, the extra arginine that gets accumulated inside,
Mhm.
it stays inside or it leaves at some point, or
No, it stays inside. The arginine concentration in the tumor microenvironment goes down because it just sort of sucks up the arginine. This might also be another reason why the tumor cells do this. They suck up the arginine because, back to that observation I told you, the arginine activates T cells. So by sucking the arginine, the tumor cells are defending themselves against activation of T cells in the tumor microenvironment.
Hi, Michael. Very nice talk.
Thank you.
So I have two doubts. So in the first part, I wanted to ask that, do you see arginine as a positive regulator of cancer cell metabolism in other tissues? And if not, what is so specific about the liver that it's only a positive modulator there?
This is seen, we have not looked directly, but you can go to databases, data sets to see whether arginine specific effects are seen in other tissues, transcriptional effects, make a correlation, in other words. And we see that this mechanism we've found is found in other tissue, in other cancers, but relatively few, maybe a small handful of other kinds of tissues. I think lung and some types of prostate, I think, but relatively few. Since that's not the case, why not? Why is it not common to all? We don't know. Other cancers have their own ways of reprogramming metabolism. There's another possibility. You can also think of this, why does this mechanism exist at all? I mean, it's not a mechanism that evolved to cause cancer at some point. Why does it exist at all? And we wondered about that. And we looked at, again, by correlation, we found that, for example, high RBM 39 expression, high ASM, agmatinase and ARG1 expression, changing in the appropriate way. We found that this also occurs in fetal liver. So I think what happens is that cancer, for those of you who don't know, is usually a dedifferentiation. Cells, when cancer cells become cancer cells, they dedifferentiate. They go back to an early dedifferentiating, highly proliferative state. And so I think this is just, it's gone to a evolutionary, a developmental early state of a liver, which is probably does not exist in other kinds of tissues.
And for the second part, for the diabetes-induced neuropathy, do you see, like, if you feed mice on a high sugar diet, does it deactivate mTOR-C2?
Sugar, no. Sugar. Well, it's a huge literature, and we always find something that supports one view or the other. But no, I would say, if I had to make a general statement, I would say high sugar will not induce TOR signaling, at least not directly, maybe through some indirect mechanism in some context, but generally no. Sonia?
Mike, I was wondering what you thought the sensory neurons were sensing and how that was controlling insulin sensitivity.
Yeah, why they're there. I'll tell you what Tim Barton is, this guy who a long time ago saw. When he saw him, he also had to propose a function of these sensory neurons in fat. What he proposed, they are a negative feedback on the sympathetic nervous system. So the sympathetic nervous system is activates the fat in response to stress. Cold, for example, activates high lipolysis in the fat to generate heat, things like this. But you have to turn that off somehow. And maybe this sensory nervous system senses whatever has to be sensed, that the temperature's gotten to be too much or enough, and or all lipids have been depleted or something, has to signal back to the brain to say, shut off the sympathetic input into the fat. But that doesn't really, it could still be doing that. But we now think it's doing much more than that. Now it's signaling not only back to the brain to signal back to the fat as a negative feedback, but more it's signaling to other tissues to control insulin sensitivity and to beta cells to control insulin production. But this is a whole new field now to understand these processes.
Here.
Yeah. Okay.
So you think RBM39 is a sensor of arginine? In other words, if I downregulate RBM39, do I see downregulation of uptake of arginine?
We do see a reduction of uptake of arginine when we reduce RBM39, but that's because we block this positive feedback loop. The RBM39 activates asparagine synthetase as part of the metabolic reprogramming. So if we eliminate RBM39, we eliminate asparagine synthetase. There's no asparagine there's an antisolute to bring in arginine. So it's via this feedback loop. But yes, it happens.
Okay, before we close, maybe a final question.
Related to the question you asked, the calcitonin-related peptidergic system,
Yeah.
there in many other contexts.
Yeah.
So are there similar feedback onto those neurons, or?
Well, we have to start looking at other systems, other neurons now. Yeah, that's why I'd like to look in the fingers, for example, and the toes in these diabetic patients. It just opens up new horizons to look at. That's why I'm so excited about it. And this is why I'm starting a second lab now in Roche to address these things.
Okay, so on that note, we'll close. Let's thank Mike once again for a wonderful talk. Okay, good afternoon, everyone. Let's get started with the second of the lectures for today. It's a great pleasure to introduce Professor Reinhard Fassler from the Max Planck Institute for Biochemistry at Martin Street in Munich. Reinhard initially trained as an MD in Austria, and after working for a while in Zimbabwe, he went to the Whitehead Institute to do a postdoc, and thereafter returned to Germany. He's been working for a long time on multiple aspects of integrin signaling, which is what he's going to talk to us about today. He's obviously a very highly decorated scientist. I won't go through all of the details, but he's a member of for example, the German National Academy of Sciences. So Reinhard, we look forward to your.
So Raghu, thank you very much for the introduction, and also I'm quite happy, I have to say, that I can be here. I was here in 2006, and it's amazing how the campus has changed since then. It's completely different. I didn't recognize it anymore. So as Raghu said, I have been working over the last 30 years on the machinery that is used by cells to attach to the extracellular matrix. And as you can see here in this slide, these attachment sites are plugs. And these plugs can have different size depending on where they are localized. They can be small, they can be large. Then they can also have a different shape. So here they look elongated, but in some cells they can be round, podosomes, in vadosomes, in cancer cells. what is common to all of them is that they are linked to actin. What is also common to them is that they have one protein which is around which all these other proteins that are recruited to these plugs, and this protein is called integrin. And integrins, just to make everybody oriented and that you can see how they look like, is these integrins are heterodimeric molecules. They consist of 2 subunits, and they are associated in the ectodomain at the head domain, as it's called. And this head domain is made by the alpha and the beta subunit. And this head domain has a small fold. And this small fold, this pocket, is the binding site for the ECM proteins, so collagen, fibronectin, and so on. There are a huge number of integrins, 24 in total. And they have different receptor ligand specificity and so on. But I will today not go into details of one integrin, because what I want to tell you is a principle. We found that what I show you today happens with all integrins except those that are expressed in blood cells. They have specific integrins. So these subunits have also a single span transmembrane domain, and what is also very typical for them is that they have very, very short cytoplasmic domains, and these cytoplasmic domains do not have catalytic activity. So they don't phosphorylate, they don't dephosphorylate, they don't ubiquitinate. They just have binding sites for proteins that have catalytic activity or for adapter proteins. So these integrins have a lot of special features. So they are mechanosensitive, and they can probe the biophysical environment of the ECM, the property of the ECM, and they can transmit this information into the cell. And what they can also do is they can translate this information into chemistry. So they can translate physics into chemistry. That's also, for example, a topic on which we are working, like many in the integrin field. These proteins have also a long half-life. Yeah, so they have a half-life of 2 to 3 days. But interestingly, they are only 10 minutes on the cell surface, on almost all cells we analyzed. So they are 10 minutes on the surface, they are internalized, travel through the endosomal system, and are recycled back to the surface. And the big question is, of course, why do they do this? Why they are only 10 minutes on the surface? They have also a very complicated folding in the ER. So for example, what we have recently found is that the transmembrane domains are absolutely essential so that the ectodomain, this head domain, can fold properly. And finally, I think the most interesting feature of integrins is that they require an activation step in order to bind ligand. And this activation step is characterized by an enormous conformational change which affects the entire molecule. So in the inactive state, the integrin is bent. And the hypothesis in the field is that when it's bent, the binding pocket is so close to the plasma membrane that the integrin cannot accommodate ligand. And when the integrin becomes activated, it extends. And what also happens at the same time is that it separates the transmembrane domain, the legs, the transmembrane domain, and the cytoplasmic domain. And only when this has happened, the integrin is able to bind ligand. So this switch from inactive integrin to an active integrin is a reversible mechanism. So also the active integrin can reversely switch back to an inactive one. It's a cycle. And what is critical is that 2 adapter proteins have to bind to the integrin. And some people in the community think this is important to induce this activation. And some think it's important just to maintain this activation. And these 2 adapter proteins are called tailin and kindlin. So both of them share a so-called firm domain. So firm domains are domains that are present in about 80 or 90% of protein, 80 to 90 proteins in our genome, in the mammalian genome. And they usually have a clover leaf-like fold, like, for example, kindlin. Tailin has an atypical firm domain, it's linear. And these firm domains have different subunits, and one of the subunit in both of these adapter proteins is the F3 subunit. This folds to a phosphotyrosine binding motif. And there are two tyrosines in the beta tail. And each of them binds specifically to one of these tyrosines. So it also requires additional amino acids next to these tyrosines. So the tailin binds to the membrane proximal tyrosine and the kindlin binds to the membrane distal tyrosine. So the binding of these two adapter proteins, as I said, is essential to fix this extended conformation. That's what we believe. So we discovered these kindlins in 2008. And what we found was kindlin 2 in a screen, in a proteomic screen. And what we found by data mining is that there are 3 kindlins in mammals. I think flies have 2. There's kindlin 1, which is expressed in epithelial cells, kindlin 2, which is everywhere expressed ubiquitously, except in blood cells. Blood cells have their own kindlin. It's called kindlin 3. What is also interesting is the localization of these 2 proteins. So kindlins have a firm domain, and they are binding at the plasma membrane to phospholipids. PIP3 a little bit less, with lower affinity to PIP2. So they are sliding on the membrane. So we have done single particle tracking and could see that they are sliding on the membrane, and when they hit an integrin, they bind. And if a tailin is also binding, then the integrin is fixed in this active conformation. Tailin, in contrast, does not have a lipid binding domain, a PIP binding domain, and it's localized in the cytoplasm. So it's recruited from the cytoplasm to the integrin tail. So the choreography of this binding suggests, that has shown by several people, including us, that kindlin binds first, because it's sliding and hits an integrin tail, and then subsequently tailin is binding. And when these 2 proteins have bound, they recruit the so-called adhesome. These are all the proteins that are found in a focal adhesion. And we have recently also, Claire Waterman at NIH, have isolated the adhesome and analyzed it proteomically. And we found 800 different proteins in these adhesomes. So it's a gigantic signaling hub. And this remarkable, incredible inactivation step that is so tightly regulated in cells is very, very important for the assembly and disassembly of focal adhesion, to control the lifetime of focal adhesions. So the lifetime of focal adhesion is tightly controlled and is very, very important, particularly for cell migration, but also for the second event, and that's cell division. And that's what I want to tell you today. So here in the next slide, you see a HeLa cell that has been seeded on fibronectin. So I will show you a movie now. And the focal adhesions are marked in yellow, and the actin cytoskeleton, the actomyosin, in red. And as soon as this cell is entering prophase, so it's entering mitosis, it's disassembling the focal adhesions and is rounding up rapidly. It's separating the chromosomes, and then the focal adhesions appear again. So you have a rapid disassembly of focal adhesion, and this rapid disassembly of focal adhesion allows them what people call what the community calls in the field, the mitotic rounding. And as soon as the chromosomes have separated, the mitosis has ended, so in telophase cytokinesis, the focal adhesions reappear again, and the 2 daughter cells use these focal adhesions then to separate and to move apart from each other. So what's interesting is that it's completely unknown how these focal adhesions are disassembled and assembled during mitosis. Although this is unknown, there were several laboratories that have analyzed it with tricks, and I will show you this. I will show you in a minute how they came up with information that shows how important this shape change is. But before I do this, I will just show you 2 stills of a high resolution, 2 images of a high resolution microscope picture of an interphase cell, of a flat cell that is either in G1, S, or G2 phase. And you see here the actin cytoskeleton, the stress fibers, and of a mitotic cell that has rounded. And what you can see, which was very difficult to see in the movie, is that this cell is changing from flat to round. And in addition, they form this finger-like protrusion. And all these events occur in a stepwise manner. And everything commences with a disassembly of the focal adhesions. So this is the first step that has to occur. The second step is that the membrane retracts. And the third one is that these protrusions form. And these protrusions are called mitotic retraction fibers. And these mitotic retraction fibers, they have two functions. So one function is that they form tiny, small adhesion sites, which are arranged like pearls on a string. And these adhesion sites fix weakly the mitotic cell to the extracellular matrix, to the substrate. So mitotic cells are adhering to the substrate very weakly, and that's the reason why it's so easily to detach them when a flask is pushed against the table. That's what was used in the 70s and 80s to synchronize mitotic cells. So people isolated the cells that were detaching from the matrix and did studies with these cells. A second function of these mitotic retraction fibers is that at the site where they connect to this central round cell body, they assemble a protein complex which serves as a landmark for the mitotic spindle to attach. So the mitotic spindle is attaching to this site, and this is to a large extent controlled by these retraction fibers. And the last step of this rounding is that the main cell body here is rounding up, and this occurs via two mechanisms, and they are pretty well understood. One is that the actomyosin at the cell cortex is contracting, so a sort of high Rho activity, high myosin 2 activity, and this leads to a rounding of the cell. And so in order to counteract the collapse of the cell, what the mitotic cell does is it opens fluid channels, and the identity of these fluid channels is not known. The opening of this fluid channel leads to an influx of fluid, and this leads to an osmotic pressure and to an outward force. So the balance of the inward force and the outward force gives you the perfect geometry of this soccer ball-like structure, which is called the central cell body. So how did people, researchers in the past, analyze that this shape change is important? So there are several laboratories which use tricks, because molecularly it was known, for example, I will show you now two examples, the flat cell and the small cell, how it was done. It was not known how these focal adhesions disassemble. So in order to keep them flat, they have added a weight on top of the cell. So for example, a polyacrylamide that was heavily cross-linked was put on the surface of these cells so that they were unable to round up. And what was reported was that if the cells remained flat, the spindle was not able to attach to the plasma membrane, because the spindle has a certain length, and this length cannot, this is limited. But if the cell is flat, the longitudinal axis of the cell is longer than the spindle, and the spindle doesn't find the attachment site. So this, the consequence of not properly attached spindles at the plasma membrane is that the chromosomes cannot be separated properly. The other problem was that the space in Z direction was confined. If a cell is flat, there's of course much less space than if the cell is round. And if the space is not sufficient for the spindle, it's also unable to find the kinetochores. So the kinetochore microtubules have a problem to find the kinetochores to attach, and then the following would be the separation of the chromosomes. But as I said, this is not possible because they were improperly anchored. So there are two consequences when this happens. So one is if the kinetochore microtubules are not properly anchored, this activates the spindle assembly checkpoint. And the spindle assembly checkpoint activates p53 and leads then, induces apoptosis of these cells. So many of these cells apoptose. If they don't and survive, these cells cannot divide, and the consequence is that they are aneuploid. So they are usually tetraploid. Now, there was a group that made the cells very small. And this was done by overexpressing a constitutive active rho. So the contractility at the cortex was much higher than the outward force of the osmotic pressure. And the consequence was that it was much smaller than the spherical normal central cell body. And also in this case, the spindle assembly checkpoint was activated. Cells underwent cell death or were aneuploid simply because the spindle did not have enough space to find the kinetochores, to attach to the kinetochores, and to separate the chromosomes. So this indicates, I think very clearly, that it's absolutely important to have the perfect geometry in order to separate this and to prevent cancer. There was a postdoc coming and joining my lab. His name is Nanpeng Chen. I will show a picture at the end. And he wanted to analyze this. He came from a lab in Heidelberg. He did his PhD in Heidelberg, where he looked at cell cycle. And he noted that nobody in the adhesion field has ever analyzed how these adhesion sites disassemble at the entry of mitosis. And he thought, he can do this in my lab. And I found this a fantastic idea. At first, I didn't believe him that nobody has looked. But after we searched for 3 days, the whole literature, we came to the conclusion it's indeed not known. And so the question was, how do we start this project? And we did a very simple experiment which worked. And so what Peng did was he synchronized cells with a double thymidine block. So the way how this works is that thymidine is added to cells, and this inhibits cells in S phase because thymidine cannot be incorporated into DNA during replication. And then the thymidine is washed out, and in order to be sure that 100% are arrested in S phase, the thymidine block is repeated. That's the reason why you do it twice, double thymidine block. And then it's washed out, and this releases the block, and the cells go into G2 phase and then into M phase. He collected cells and prepared lysates at different time points after the washout, after the release of the block. Yeah, so he isolated at time when he did the washout, 4 hours later, which was S phase. So this was very well established in HeLa cells. These were HeLa cells which we used in 7 hours in G2 and 9 hours in M phase, and then 13 hours after the release, the cells were back in G1 phase. And we analyzed around 30 focal adhesion proteins, of which we thought in our lab, these are very important focal adhesion proteins because we determined the adhesome. Remember, we did, of course, not look at all 800, but we took about 30, of which we are convinced that they are absolutely essential for integrin function, and looked whether they change in abundance. That was the idea. So a relatively simple idea, and to our surprise, it worked. And we found that none of these 25 except 2 proteins indeed changed abundance. And we couldn't believe it when we saw these plots that it was kindling. Yeah, this is a protein which we found quite some time ago, and now we find that it's decreasing in abundance in M phase cells. And this was carried out not only in HeLa cells, and I think he analyzed about 10 different cell lines, even primary cells. It occurs in all cells, epithelial cells, neuronal stem cells, all cells in our body that are not blood cells are undergoing mitotic rounding in M phase. And in all cells we analyzed, we found that these kindlings are decreasing. What we also noted was that the kindling is changing, the remaining kindling that is not degraded is changing the apparent molecular weight. So we saw an upshift of the kindling protein. So the decrease is dramatic. So it's about 80%. So we did densitometric measurements, and you can see that the kindlings, this is the red lines, kindlin 1 and 2, they are decreasing by around 8%. So 8% of the protein is lost abruptly. Yeah, so this focal adhesion disassembly at the mitotic entry occurs in very few minutes. So it's extremely fast. And what we also noted was that another protein is changing its apparent molecular weight. It's called paxillin, but the abundance didn't change. So what we did next was we wanted to see where are these remaining 20% of the kindlin that are not degraded, so that have this upshift. And we stained cells with an antibody against kindlin, and we also overexpressed an EGFB kindlin. So we did a lot of different experiments. In all of them, what we found was that in interface cells, kindlin, as expected, is in focal adhesions. It's co-localizing with an antibody that stains activated integrins. So integrins have different epitopes, and there are epitopes that are masked when the integrin is bent. And this 12G10 epitope is only available for the antibody when the integrin is extended. So this fits perfectly because here the integrins are active in the focal adhesions. They are also linked to F-actin. And when we looked into mitotic cells, we found that the remaining 20% are diffusely more or less distributed in the central cell body. And when we looked at long exposure onto these mitotic retraction fibers, we found that these mitotic retraction fibers had tiny adhesion sites, which were very difficult to visualize. And they also co-localized with this 12G10 epitope. This can be very nicely seen here also in this line scan. And they are arranged, as I said in the introduction, really like pearls on a string along this actin filament that is in these retraction fibers. So we see them in the central cell body, the remaining kindlin, and we see it in these tiny adhesions, which are so important to weakly attach the mitotic cell to the ECM. So next we analyzed the upshift of kindlin. So we wanted to know, why do these remaining 20% kindlin change the apparent molecular weight? And this, so we were pretty sure this is due to modification, of course. Everybody would have assumed this. And we hoped that it's phosphorylation, because that's easy to analyze. And in order to test whether it's indeed phosphorylation, what we did was, so here we synchronized the cells, just that you know the experiment, how the experiment was designed. We synchronized our cells with an inhibitor against kinesin, a specific kinesin that is important for the spindle formation. So if the spindle doesn't form, the cells are stop in mitosis. And you can see that the kindlin abundance is decreasing, and we see the upshift. And then we took the same lysate, an aliquot of the same lysate, and treated the lysate with phosphatases. So alkaline and lambda phosphatases. And what we then saw was that the upshift is reversed. So we see the normal molecular weight of kindlin, which is around 70 kilodalton. And this indicates that these remaining 20% kindlin, which are not degraded, which are not decreasing in abundance, which survive in mitosis, these 20% remaining kindlin are phosphorylated. So this was then followed by a phosphoproteome analysis. So these here, we found about 25,000 phosphopeptides, both in mitotic cells. These are these here, the phosphopeptides of mitotic cells, and also in interface cells. But of course, the phosphopeptides were different. And one difference was in kindlin 1 and 2. So these were very high hits, which were not present in interface cells. So we found this phosphopeptide in which kindlin 2 was phosphorylated at a very specific serine, only one serine, 818, and kindlin 1 was serine 179. And when we tested, or when we looked where these serines are localized, what we found, it was localized to a small, unfolded, unstructured, flexible region, which is in the firm domain. And the site is conserved, also in Drosophila and C. elegans, in all species we analyzed. And what we also found was that the serine is followed both in kindlin 1 as well as in kindlin 2 by a proline. And if it's followed by a proline, this indicates that it's a proline-directed phosphosite. And proline-directed phosphosites are recognized by CDK1, cyclin-dependent kinase 1. So our hypothesis was that the reason why it's upshifted is because it's phosphorylated at the specific site in this unfolded, unstructured region. And this phosphorylation is occurring through the CDK1 cyclin B1 complex. So this is the master kinase of mitosis. Okay, so what we did next was we confirmed this, of course, with a lot of experiment. And I will just summarize this here. So what we did was we expressed recombinant and purified recombinant kindlin 1 and 2. So this is easy, can be expressed in bacteria, can be purified in high amounts. And we also expressed and purified, that's done in our core facility, CDK1 and cyclin B1. And we did in vitro phosphorylation. And then we went to the mass spec. And we found that also in vitro, when we phosphorylate with these recombinant complex, we find exactly the same phosphosites. What we also found, particularly in kindlin 2, was that also adjacent serines were phosphorylated, but to a much, much lower extent. And this has also been reported for other CDK phosphosites, that adjacent serines and threonines can become phosphorylated. What we also performed was co-immunoprecipitation experiments. So we precipitated kindlin 1 and 2 and could find that kindlin 1 and 2 co-precipitate CDK1 and cyclin B1. And so finally, what Peng also did was he made an anti-phosphospecific antibody. So we synthesized this peptide in our institute, had a phosphosite at serine S181, and it was immunized in rabbits. And this antibody works perfectly. So here are cells that have been, again, inhibited in mitosis using this kinesin inhibitor. We see the decrease in kindlin 2 and the upshift. And this phosphor-specific antibody recognizes this band. And of course, also phosphohistone 3, this indicates that the cells are in mitosis. When we treat now these lysates with phosphatases, what we see is that the upshift is decreased, and this signal of the phosphor-specific antibody is lost, also the signals of the phosphohistone 3, because the phosphatases also remove the phosphorylation of the histone. And this antibody also works well in immunostaining. So what we could do is we could stain our cells, and we found that in interface, there is no signal, because in focal adhesions, in interface, the kindlin is not phosphorylated, so this antibody also doesn't recognize the kindlin. When cells enter mitosis, we see a signal. The signal peaks in metaphase, is decreasing in anaphase, further in telophase, and in cytokinesis, it's gone again. And the mitotic retraction fibers, which have these little tiny adhesion sites, they are also positive for our kindlin phosphor-specific antibody. So these 20% of the kindlin are in the central cell body and in these sites, and this can also now be shown with this antibody. So what Peng did next was he wanted to know what is the consequence of this phosphorylation. And so the way how he analyzed this was by introducing point mutation into the serine 818 of kindlin 2. So he substituted the serine for a phosphomimetic amino acid. So he used an aspartic acid. And he also substituted it with a phosphoinhibitory amino acid, which is an alanine residue. And he also made a kindlin in which all these additional 4 amino acids, which were also phosphorylated in vitro, we didn't see them in vivo, but in vitro, were also substituted for alanine. He called this the 5SA kindlin 2. So we took our cells that lack kindlin 1 and 2, and we expressed these wild type kindlin 2 and these mutants in this cell line and looked for stability of kindlin 2 in interface cells. So not in mitotic cells. We analyzed this in interface cells. And we did this by performing a cyclohexamide assay. So cyclohexamide inhibits protein translation at the at the ER, and the remaining pool of kindlin can then be followed by immunoblotting over the time. And wild type cells, wild type kindlin, which is the black line, has this degradation rate in interface. And when the serine 181 is mutated into alanine, or these 5 were mutated into alanine, we have the same degradation rate as the wild type kindlin. However, when the serine 818 is substituted for an phosphomimetic aspartic acid, what we see is a rapid degradation of this kindlin, also in interface. So what these data indicate is that if the kindlin would be phosphorylated in interface, but remember, it's only phosphorylated by the mitotic kinase complex. But if it would be, it would also be degraded in interface. And the only conclusion which one can draw from this experiment is that the degradation complex, which degrades kindlin in mitosis, must be present also in interface. So the degradation complex is present all the time. But it only is only going after the kindling if it's phosphorylated, and that only happens in mitosis. What we also did was we inhibited the cells with either buffelomycin, which inhibits the lysosome, or with MG132, which inhibits the proteasome. And these experiments revealed that only the inhibition of the proteasome rescued the kindlin levels in these interface cells. Buffelomycin did not. And these data indicate that the phosphorylation of this serine here is probably a phosphodegron, and this phosphodegron is probably the trigger to ubiquitinate the kindlin, because only ubiquitinated protein are degraded by the proteasome. So our idea, our hypothesis now was we have to look for the degradation complex. But before we did this, he also made movies of these mutant cells. And you can see here these wild type cells, they round up in mitosis, they separate the chromosome, make 2 daughter cells, and become flat again. The other cells, which carry these mutations, behave very differently. So the cells which carry the phosphomimicking mutation, this aspartic acid substitution, they are smaller already in interface. They rapidly, much faster than a wild type cell, round up in mitosis, but they are unable to separate into 2 daughter cells. And if they do, they are aneuploid. So they either die or become aneuploid. Cells which have the alanine substitution, they remain flat. You can see this probably here. The cell tries to round up, but it's impossible because the focal adhesions are not disassembled. And the consequence is that these cells are also either tetraploid, or here's an example of a cell that goes into apoptosis. So in these cells, the spindle assembly checkpoint is most likely activated, and that leads to the killing of the cell. And you can see here the reasons, because these cells, here you have a still image of these movies, and you can see in metaphase, you have in wild type kindlin perfect spindle that attaches in the metaplate to the chromosomes. The cell is perfectly round, and if the serine is substituted for alanine, then the cell has spindle defects, has centrosomes defects. As I said, activates the spindle checkpoint, has increased rate of apoptosis or multinucleation. And the reason is because the cell is large. The cell cannot attach the mitotic spindle at the plasma membrane, and furthermore, due to the confinement, the spindle is also unable to attach to all these chromosomes, and therefore it's not possible to align them. And the same happens when the cell is expressing a kindlin that carries this alanine mutations, this phosphomimetic mutation. The cell is very, very small, which I showed you in the introduction. The defect is identical. So either to be small or to be flat, the mitosis does not work. So what I showed you so far is that the CDK cyclin B1 complex at mitotic entry we know it's binding through cyclin B1, it's binding to the kindlin, is phosphorylating the kindlin at this unstructured loop. The phosphorylation leads to degradation, to proteasomal degradation, focal adhesion disassembly, and mitotic grounding. And the question is, how does it degrade this kindlin? And so what we did was, as I said before, we assumed that it's a ubiquitination event that leads or triggers this degradation. We were looking for E3 ligases. And the way how we did this was we took our cells that express a EGFP-tag kindlin in which the serine was substituted for an aspartic acid, because this kindlin is degraded in interphase. Remember, it's rapidly degraded in interphase, so the EGFP levels are low. And so now what we did was we screened these cells with an siRNA library against E3 ligases, E2 enzymes, E1 enzymes, and adapter proteins that recruit E1, E2, and E3 enzymes. So around 1,100 siRNAs. And so our hypothesis was that if the E3 ligase is depleted, then the kindlin should not be degraded in interphase, and the EGFP levels should rise, and we should see this when we screen these cells. And so we did this, and we found indeed two hits that were immediately capturing our attention. So one was CALI9, and the other one was an FBOX protein, which is called FBXL10. So CALI9 is interesting because CALI9 is an adapter protein. I will show you a cartoon how it works, that has been analyzed genetically in mice. And what has been found was that these, so there was a group in the US that made a knockout of CALI9, and if CALI9 is not expressed in mice, these mice survive, but develop cancer. So they all develop spontaneously cancer, and this could of course be, if this CALI9 is responsible for the degradation, that the cells remain flat, the spindle does not perfectly attach, does not separate the chromosomes, and the consequence is that these mice may develop aneuploidy, and this leads then to the cancer which is seen in these mice. So CALI9 was a very interesting candidate for us. FBXL10 is a protein that is not well studied. So there are only 2 papers which we found, and one showed that it's a histone methylase, so a lysine demethylase 2B. It demethylates histone 3, and another paper showed that it's involved in epigenetics. It's however expressed both in the cytoplasm as well as in the nucleus. And the reason why we thought it's a very interesting hit is because calins have a banana-shaped form. So there are in total 8 calins, and they all look like this here. So this is this banana shape, and they have at the end terminus a binding site for an adapter protein that is called substrate adapter. And this substrate adapter binds a substrate receptor, and this is very often an FBOX protein. That's the reason why we were so excited. And this FBOX protein, this substrate receptor, binds the substrate, which is in our case, the kindlin. At the C terminus, these calins bind an E3 ligase, and together with the help of an E2 enzymes, they transfer ubiquitin chains on the substrate. And these ubiquitin chains, if it's a 48 linked ubiquitin chain, leads then to binding to the proteasome and to the degradation of the protein. So that's the reason why it was a very interesting hit for us. So we did a lot of experiments, and I will only show you two before I end. And so this here is an experiment in which Peng did an immunoprecipitation of Calin-9. And by immunoprecipitating Calin-9, he immunoprecipitated the F-box protein and the substrate adapter, which we don't know. So he had the entire complex, and together with at the C-terminus, the E3 and the E2 enzyme. So this complex was precipitated. And what he then did, and you can see that the F-box protein is well precipitated with Calin-9, and then he added Kindlin to this precipitate. So either he added Kindlin that was untreated, so wild-type Kindlin recombinantly expressed and purified, or he added Kindlin 2 that was in vitro phosphorylated with CDK1 and cyclin B1. And in addition, he also added ubiquitin, either wild-type ubiquitin or a ubiquitin which carries a substitution at lysine 48, which is making this chain of ubiquitins that are recognized by the proteasome. But if this lysine 48 is substituted for an arginine, this is not possible anymore. And what you can see here is that when he added the wild-type Kindlin, the non-phosphorylated Kindlin, to the immunoprecipitate, and then blotted also a wild-type ubiquitin, and then he blotted for ubiquitin, the Kindlin is not ubiquitinated because it's not phosphorylated. It cannot bind to the Calin complex. And when the Kindlin is in vitro phosphorylated by the CDK cyclin 1 complex, you see the second band, the upshift. And then when this is added together with a wild-type ubiquitin to this precipitate, the Kindlin is highly ubiquitinated. And when the ubiquitin carries this lysine to arginine mutation, is phosphorylated, it's not ubiquitinated. There's one ubiquitin added, but not the chain. And the consequence is, of course, that this indicates that this complex indeed ubiquitinates the Kindlin in M phase. And so what Peng also did was he did knockdown experiments of Calin-9 and of XBL10, and he sees exactly the same results as if the Kindlin serine 181 is substituted to an alanine. The cells remain flat because in the absence of the degradation machinery, the phosphorylated Kindlin can now not be degraded. And the consequence is that these cells have also spindle defect, they have centrosome defect, they cannot attach to the chromosomes, they cannot separate them. And basically we see the same either aneuploidy or apoptosis due to the spindle assembly checkpoint activation. So the summary basically of this story, what we found is that the Kindlin is at mitotic entry bound by this CDK cyclin B1 complex. This leads to the phosphorylation, and then the phosphorylated Kindlin is recruited to the Calin-9 FBXL10 complex, and this complex then adds a ubiquitin chain, a 48-linked ubiquitin chain to Kindlin, and this leads then to proteasomal degradation, to the disassembly of the focal adhesions, and this initiates the rounding of the mitotic cell. Okay, so these experiments which I showed you today, they have all been done by one person basically. So he did 95% of the experiments, and this was Nanpeng Chen, who joined, as I said, from Heidelberg. He did a training in a cell cycle lab, and Jonas Aretz, he's a chemist in our lab. He prepared the recombinant proteins, the Kindlin 1, Kindlin 2, CDK1, and cyclin B1. So this is it. So if you have interesting questions, challenging questions, I have still a few more slides. I didn't put them into my presentation because Raghu told me that it's only 50 minutes we have time, so I had to take some slides out. But if you have interesting questions, I will show you them. Thank you.
Thank you, Reinhard. Questions?
Great talk. So I want to ask, what protects that 20% phosphorylated kindlin from degradation when it's forming those thin protrusions during mitosis? Why is that 20% preserved?
Yeah, so that's a question which we were also addressing. So what we found was that these little dots, they are positive for FBXL10. So this phosphorylated kindlin, which is in these small adhesion sites, in these retraction fibers, these mitotic retraction fibers, recruits FBXL10, but Cali9 is not there. So we don't know why it's not there. So either it's transported via microtubules, because there are also microtubules, or maybe it's a large complex and cannot enter these tiny, thin retraction fibers. We don't know, but that's the reason. It's not recruited to these sites.
Maybe I'll ask a question. So you mentioned at the beginning of the talk the strategy of removing the integrins from the surface by endocytosis. And this appears to be a strategy to inactivate their function during the cell cycle. So are these used separately at different points, or is there some strategy to which one you choose for which point?
So integrins are continuously recycled. Yeah, and the reason why they are continuously recycled, and at the early endosome, there's a decision center which decides whether they are routed to the lysosome, whether they are recycled back. And there are two reasons why they are probably recycled so frequently. One is that integrins, when cells move, for example, integrins which are in the rear are internalized and shuffled to the front where new focal adhesions are formed, because in the rear they are disassembled and in the front they are assembled. So that's at least has been shown by a group at the Curie Institute. What we could show is that integrins recruit in early endosomes a large complex consisting of sortin, nexin, 17. So this is a specific sortin. And this in turn recruits the retriever complex. And this prevents integrin from routing into lysosomes so they can go back. And our hypothesis is that they check in early endosomes whether integrins are still properly functioning, properly folded or not. Because if an integrin ectodomain becomes unfolded due to the actomyosin pulling forces, it cannot refold in endosomes because there are no chaperones. It requires chaperones for folding. And you have to sort, the cell sorts these integrins out, and we assume that this is done by the sortin nexin 17 retriever complex.
So given this high rate of turnover of integrins from the cell surface, integrins are also used in very stable structures, for example, in myotendinous junctions, where I would have thought that their continuous presence as attachment molecules or interfaces are important. So what's the mechanism of turnover there, and how do you maintain the stability of attachment while ensuring turnover?
So the binding of a ligand to an integrin is dependent on the force which the integrin ligand bond experiences. So if the force is very low, they make so-called slip bonds. Yeah, and these slip bonds lead then to the disruption of the integrin ligation.
Right.
If the force is very high, and this is the case in myotendinous junction, they form a so-called catch bond. So this is a way to increase the lifetime of the bond. But the lifetime of a bond is always in seconds. So it's maybe in milliseconds if it's a slip bond, and it's seconds if it's a catch bond. So what we have done together with a group in France, we have done single particle tracking. And what can be seen when single particles are tracked, that integrins enter focal adhesions, stay there for a few seconds, get out, get in again, get out. So if you look only at the single integrin, and the same occurs also for kindlin. Kindlin is sliding at the membrane, gets into the focal adhesion, stays there for a few seconds, gets out. and the tailing comes from the cytoplasm. So there is an increase in lifetime, and there is a second mechanism which helps. But we are always talking about seconds to minutes. The second mechanism is that not only the lifetime of the bond can be increased, but also the lifetime of the adhesion, the whole adhesion, and that's done by avidity. Yeah. So in the extracellular space, there are, let's say, fibronectins. Fibronectins are fibrils. In the case of the myotendinous junction, it's a basement membrane. So you have a lot of laminins, and if one integrin falls off, it immediately finds another RGD of fibronectin to which it can bind. And this avidity is also increasing the lifetime. But just be aware, but it's probably important because, you know, in such a focal adhesion there are 1,000 integrins. Yeah, so altogether, this is probably the strength that they provide, or maybe also in, you know, in a myotendinous junctions, you have probably 10,000. I have no idea. I've never looked at this. We are only working with focal adhesions. And I think that's the reason why it's so stable. So you need high force to get a good bond, a catch bond, and the avidity that's there all the time is increasing this lifetime.
Hi, thanks for the talk. I wanted to ask that, especially in the case of the phosphomutants, is it just the size of the cell that is dictating the defects in mitosis, or does a phosphomimetic or a non-phosphorylatable variant have other downstream signaling processes that lead to the defects in mitosis that you see?
Yeah. So what I can tell you is that so the phosphomimetic mutant that is degraded in interface because the complex is there. You know, Calin-9 is there, FBXL10. It makes all sense, you know, that it's degraded. So this flexible domain, if this is removed, interface cells behave normal. They adhere normal, they spread normal, they migrate normal. If this, because the loop can be cut out, and you can express such a mutant in cells. What we could also show is that the binding to the integrin, the affinity, is not changing, which is not surprising because it's a completely different domain. You know, the firm domains have subdomains that are linked via linker peptides. So if you express only one subdomain, it's perfectly folding, doesn't require the other ones. So affinity to the integrin tail is not affected. If this loop is removed, interface cells move, they spread, everything is normal, but as soon as they enter apoptosis without this loop, they have the same problems. Yeah, so that's, I think it's no other function. It's a mitotic specific function.
And you mentioned in the beginning that Paxillin also, the band of Paxillin also moves up. So have you?
That's a very interesting question. Because this question I don't get very often. So this is interesting because, you know, I told you in the introduction that it's a stepwise mechanism that leads to the rounding. And so basically what our data show is that we know now how focal adhesions disassemble. Other people have shown how the rounding occurs, you know, with this balanced force, inward force and outward force generation. But it has not been shown how plasma membrane retract. So, you know, when we started this project, I always wrote plasma membrane retraction dash collapse, because our idea was that if the focal adhesions disassemble, it automatically collapses. But this is not the case. I can show you here just maybe one experiment. So the question which we addressed is rounding is essential, because if you make the mutation, what happens is that you have all these defects. But is it sufficient? So is it a collapse? Or does something else is still on top of this degradation required? And so the way how we addressed this question was we took interface cells and decreased the kindlin level to 20%. So if kindlin decreased to 20 would be sufficient for rounding, then these cells should round up. So they are smaller, they don't spread so well because they have less kindling, but they are still flat. And then, of course, we thought this could be because of paxillin. And interestingly, when we take cells that have no paxillin but 100% kindling, they are flat, they have no phenotype. That has been reported by many people. But if these cells have only 20% kindling and have no paxillin, they round up. So there are two events happening. On one hand, kindling is phosphorylated, which kills the focal adhesions. And the second is paxillin is phosphorylated, and this does something. And paxillin is famous for RAC activation. So there are 4 complexes that are recruited to paxillin in order to activate RAC up to 3 and protrusion. So our hypothesis was that the phosphorylation, and we know now it's also CDK1, cyclin B1, phosphorylates paxillin, and that leads to the inactivation of this pathway. And then you have everything. So then the cells have no focal adhesions, no RAC activity, no protrusion, so it collapses, and the cell has then just around the central cell body. And we analyzed this, and we found that 20% cells with a RAC inhibitor round up, but not 800% kindling. 20% kindling with an ARP2/3 inhibitor also round up, but not cells which have 100% kindling. And then we analyzed all these different complexes based also on proteomics. We made proteomics and found that there is one complex that does not bind. All the others bind. And that's this one here. The complex that is recruited to LD4, it's an adapter protein complex consisting of GIT, NCK, beta-PIX, and PAC. PAC is a kinase that activates GAFs. And this leads among all the others can also do activate RAC, but this is the only one that was not bound to the phosphorylated paxillin. And it's indeed so when we remove GIT, PIX, PAC, NCK, and have 20% kindling, they also round up in interface. So what we believe is that CDK does 2 things at the mitotic entry. It is responsible that the focal adhesions are dismantled, absolutely essential. If this does not occur, it's a huge problem. And what we also found, this is a paper we are writing up now, that paxillin is phosphorylated, and the phosphorylation of paxillin leads to paxillin inactivation, not degradation, inactivation. It cannot recruit anymore a complex. And this complex is so important for the membrane protrusion. So both together. So I think that the story is now clear. So the whole pathway, the whole choreography, I mean, this is a real, it's like in a ballet. You know, you dismantle first the focal adhesions, then the retraction is not working anymore. The cells then probably collapses indeed, and then the rounding works, and then at the end you do the ballet is again making focal adhesions, the cell becomes flat again and separates the daughter cells. So I think it's magnificent. I love it, this story, because it's also so important because, you know, there is a lot of antibodies that are used for cancer treatment against integrins, and it may work for metastasis, but it may not work for the primary tumor because you probably also cancer cells, by inhibiting the rounding, induce more mutations, and I think this treatment aggravates cancer.
Yeah, it's a nice talk. I'm just want to know how Rho kinase in working hands on hand when the disassembly of focal adhesion happen, because Rho activation also need when circulation of the cells.
Mm-hmm.
So how is the dynamics this happening?
So this has been studied by other groups, and what has been shown and published is that the stress fibers are not, so usually when a cell migrates, stress fibers are also disassembled into G-actin. But at the entry of mitosis, it's not clear exactly how it works. The stress fiber are cut into pieces. And these pieces are transported to the cortex. So usually at the cortex is a branched actin network. But in mitotic cells, it's a filamentous actin. And this filamentous actin contains, of course, myosin 2 motors and so on. So they work at the cortex like stress fibers. But they are not straight, like in a flat cell, but they are surrounding the entire inner leaflet of the membrane. And when myosin 2, so ROROC, is activated and myosin 2 is activated, then these stress fibers, if you wish, at the cortex are contracting, and that leads then to the rounding. So that has been very well analyzed. Also, people have shown, as I said, they don't know how it's cut into pieces, but they have shown in movies how the cut filament pieces are translocating to the cortex. Also, the machinery that translocates them is not known, but you know, the scenario is known how it works.
Hi, that's a very interesting mechanism. I'm thinking more from a therapeutic angle. You know, there's a lot in the biologic space, and the small molecule drug discovery is also picking up for particularly RGD domain containing integrants. And most of them are directed towards AV beta interacting with respective extracellular matrix proteins. Now, how do you position kindlin as a target? You know, and if you're saying the firm domains are unstructured, and if you want to develop a PROTAC or a molecular glue or small molecule such that it aids its degradation in a more controlled fashion, how can we move forward with your target as a therapeutic in the therapeutic space?
Yeah, so I would use kindlin inhibitors as a or kindlin as a target only in cells that are post-mitotic, of course. But if you use an inhibitor that targets post-mitotic cells, it will probably also target mitotic cells. And that's the problem. So I can just tell you one example. So Merck in Germany has made a huge phase 2 trial with patients in which they treated glioblastoma patients with cyclic RGD. So that inhibits the integrin. It doesn't matter if you inhibit the integrin or if you inhibit kindlin. You just have to inhibit or inactivate the integrin. If you inhibit kindlin, that leads to the integrin activation. Remember, it's tailin and kindlin that leads to this activation or maintains this activation. If you have one, if you lack one, we did this in cells in mice, the cells are in suspension, they cannot adhere. So it can be the integrin, it can be the kindlin. So they treated, they inhibited the integrin of glioblastoma patients. And based on studies which were published by Cheresh in San Diego. So he showed in Corin Alantuis membrane experiments that all kinds of tumors, when he treated them with cyclic RGD, he could inhibit growth and also movement, also so invasion. And the problem was that the patients that were treated with the RGD became worse. And those that were treated MOC, they did better. And so I could imagine that, that it could be that these cancer cells became even more aggressive if you remove the integrin. So I think it's extremely dangerous to do this. I know it's done in many, or people think about this because they think about metastasis. And I think in metastasis it may work, but it may not work in the primary, or you know, a migrating cell does not divide, because remember, if it rounds up, there's no way that you can move. And a cell that divides does not migrate. So, but a primary tumor, which is hit by anti-integrin agents or anti-kindlin or anti-tailin agents, that can only become worse. So I would myself never treat with an integrin inhibitor or with a kindlin inhibitor.
Thank you.
Thank you. Final question, Vanupo.
So I have a question on the spindle organization. Does the spindle defects in the spindle organization comes from the integrin disassemblies? Is it a direct, only a mechanical impact of not being able to mitotically round up, or there is a biochemical part to disassemble or not correctly assemble the spindle fibers?
So our hypothesis is that the spindle is perfectly assembled. There's no defect. our hypothesis is that the spindle is just not attached, yeah, because it's not possible to increase the length of a spindle. A spindle has always a constant length. And if the cell is longer than the spindle, it cannot attach. So that's one problem. And we think the second problem of this normally assembled spindle is the confinement. That's, I think, a huge problem. You know, a spindle, the kinetochores, microtubules, they need space in order to find the kinetochores. And if the space is not there, they will not find it. That's a problem. So it's not, I think the spindle is normally assembled.
Okay, I think we'll close there and thank Reinhard for a wonderful talk. And okay, so welcome to the last of today's 3 talks. Our speaker is Vivek Malhotra. He has a green card to enter NCBS. And he doesn't need an introduction, or he'd like to introduce himself. But Vivek is from the CRG in Barcelona, though he spends a lot of his time here and is planning to spend more time here. But jokes apart, you know, if you look back at all of the papers that he's published over the years, you'll see a certain elegance to the way he develops scientific stories. And he's been involved in many important discoveries in the understanding of how things are moved around in cells from the time he was a postdoc with Randy Sheckman.
No,
oh, sorry.
Please. Rothman,
sorry, yeah. Rothman. It was close.
No.
And so he's worked out a lot of important things about processes that control movement of cargo in cells. But in the last few years, he's been looking at some very unusual ways of moving cargo in cells. And that's, I think, what he's going to talk about. He's promised to talk about very new things.
Well, so you know what happens when you, it's very simple. When you visit someplace routinely, you kind of lose that attraction that you hold. That so I come here so often that I worry that people think that I'm just a resident of this place, and so you in fact forget the details that may or may not matter. So I think this whole idea of EMBO lecture series is not so much about me giving, you know, a resume of what I do, but also to bring others into seeing how science is done in India. And I think it's very, very important because we keep saying that India has a lot of potential, and there are great institutes. Some people know it very well, others don't. And the more you visit on a formal or informal visit manner, the more you realize how much good stuff happens here. And I'm hoping that there will come a day in my lifetime when students wouldn't have to go to, you know, get degrees like BTA, being to America, or BTE, being to England. And they would go from one NCBS-like place to another place and vice versa. And I think that's the sign of success. With that little bit of background, and I think that I'm going to keep it very, very informal. So if you don't understand something, please stop me and I'll try to explain as and best to my abilities. So this is, so before Palade, I think even in the late 50s, it was very clear that cells secrete proteins. So they knew about collagens. They knew about enzymes secreted by the pancreas. And so there was this interest in trying to understand how is it that these proteins are secreted? So there was a big question. And since then, say from the 50s to now, we now know that close to 8,000 proteins encoded by the human genome would enter a compartment called the endoplasmic reticulum. And of these 8,000 proteins, the vast majority leave the ER, and of those, a vast majority are secreted. And this includes the insulins, the neurotransmitters, hormones, antibodies, collagens, mucins, you name it, okay, cytokines, chemokines. And they're very important. And it's not just a problem of secreting proteins. I think you have to worry about how you secrete them in the right quantities. So 25% of your dry protein weight is the collagens. Your bones are predominantly collagens. Skin, tissues. Skin is the largest tissue. You wouldn't have skin if you did not secrete collagens and components of the ECM. And there are 28 different genes that encode for the 28 different collagens we make. And then there are collagen-like proteins. And you secrete a liter of mucins per day. This is bucket loads. And when a B cell becomes plasma cell and starts secreting antibodies, you release antibodies at the rate of 10,000 molecules of antibody per minute. That's huge. This was not known at the time when Palade and others were trying to understand how a cell is built, how a cell is compartmentalized. So their idea at that time was to understand the path taken by a protein from its site of synthesis to final destination, which is the cell surface. So Leblanc, a Canadian, and George Palade, a Romanian, but George was working at the Rockefeller. They did a series of experiments based on sectioning tissues and doing autoradiography of cells that had been incubated with radio-labeled amino acids, and then follow incorporation of amino acids into the proteins, and then following radioactivity across the site of from the site of synthesis to the final place. It sounds very simple now because you can do it, but at that time it was it was almost impossible to envision that you could do that. And so George Palade, I'm going to turn my email off, otherwise we'll be getting, so George Palade came up with this pathway that newly synthesized proteins, especially those made in the endoplasmic reticulum, they leave ER, they come to the Golgi, and from this compartment, they were found contained in a structure which they call a condensing vacuole. This would be the equivalent of a secretory granule now. So this would be where you would put insulin and mucins and hormones, etc. Okay, and then ultimately the protein that he was looking at, chymotrypsin inhibitor in pancreatic acinar cells, was found outside the cells. So he proposed that you go from ER to the Golgi to this compartment and then out, a pathway of protein secretion. He never really saw any vesicles in the vicinity of the ER or in the Golgi area, but he proposed that this would be the right way to do it, because this way you take only the material that needs to be exported to the compartment where it has to go. So there is no mixing of compartmental content. And according to him, the compartments were solid, fixed structures. You moved items between the compartments, but there was no mixing of the compartments per se. And this is very elegant. But you know very well now that biology doesn't care for elegance. This is not some sort of fashion show. Yeah, biology cares for correct. And what is correct today, this is a lesson for all the young ones. Never mind the dogma. If you come to my office, there's a big sign. Never mind the dogma. Okay, what is correct today is likely to be not entirely correct in a few years from now, because as we learn more, we realize there are more holes in our thinking. So George proposed that vesicles are the modus operandi. This is the mechanism by which you move material from one compartment to the other. Okay? It became the so-called vesicle-mediated trafficking of cargoes. And I'm going to tell you that this is not correct. This needs a lot of revision. And cells use made-to-measure pathways for exporting cargoes. Okay? And which goes against this idea of vesicle only as the carriers for transport. So this is, of course, George's proposal, and this requires that the proteins have to enter the ER. Yeah, that was Global's contribution to biology, was that all proteins that enter the ER they contain a specific ticket for entry. We identified about 15, 20 years ago that there are many proteins that live in the cytoplasm and can be secreted without entering the ER. And that becomes unconventional pathway of protein secretion. We're still working on that, but that is not what I'm going to talk. That's not what I'm going to present today. So this concept of vesicles as the carriers of cargo then became the not the fashion, but the need to then identify these vesicles and then identify how you make a vesicle, understand how vesicles are targeted to the right compartment and how they fuse to deliver the contents, right? That's what you have to do. You have to make a vesicle at the ER, which will be filled with material that needs to be taken. It will come to the Golgi, it would fuse, deliver the contents, and you keep doing this till you deliver the cargo to its right destination. So what happened then was that groups like Randy Sheckman's and Jim Rothman's, I worked with Rothman. So Rothman took a very brave approach to reconstitute this process in vitro. Okay? And Sheckman took an approach of identifying genes required for protein secretion, but he performed the screen in Saccharomyces cerevisiae. It was very important, but it was incomplete because you might have looked at yourself recently and you don't look anything like a yeast. Yeah? Yeast do not produce collagens. They're not multicellular, nor do they produce mucins, nor do they produce insulin, antibodies. They do not have a skin per se, and they certainly don't have bones. They're not multicellular, right? So you know right at the outset that it's a limited approach in terms of what you're going to get out of it. But that was the best thing you could do in the late 60s, early 70s. So a lot of work went into this. And then, in fact, we were able to reconstitute many of these processes. And in 1989, we identified a class of vesicles called COP1, which bud from the Golgi. I, in fact, had purified these, which bud from the Golgi. And the thinking at that time, to the best of our abilities, was that they go from one cistern of the Golgi to the next one in the direction of the plasma membrane. But it turns out over the years that COP1 vesicles bring material back to the ER exit site. So they're most likely recycling rather than taking material in the forward direction. So how do you go out of the ER in the forward direction? So in 1992, Randy identified a class of vesicles, which had to be called COP2 because they were second in the list. These vesicles in Saccharomyces cerevisiae collect material from the ER and take it in the forward direction. So then it became very clear that you have COP2 vesicles going here, COP1 vesicles bringing the material back. Now, these were purified. This is what they look like. This is what COP2 vesicles look like, and they look identical to COP1 at the microscopy level. But their molecular composition is very different. In fact, it's completely different. Now, this is an important piece of work that has to be described before I launch into my talk. So Randy, Bill Balch, and many others worked for at least 2.5 decades for us to have this picture up here. So the basic tenet of the mechanism by which you make a COP2 vesicle is the following: that there is a protein in the endoplasmic reticulum called SEC12. SEC stands for secretory genes, because it came from Randy Sheckman's famous genetic screen in Saccharomyces cerevisiae. This protein has the capacity to exchange GDP on SAR1 to GTP. It's a nucleotide exchange factor. Yeah? Once these two meet, this has now GTP bound to it, and in a GTP bound form, it extends a finger, an amphipathic helix, which allows it to insert into the outer leaflet, as shown here. This protein then recruits a dimer made up of SEC23 and SEC24. They bind. The binding of this results in a reaction by which you can now start collecting material that needs to be exported. But there is a requirement that the material has to span the membrane. So this has to be a transmembrane protein which can bind to 4, so you start collecting material. This in turn can recruit cytoplasmic luminal protein, meaning proteins that lack a transmembrane domain. So you imagine insulin binding to insulin receptor, for example. Yeah. So you collect material. Now, during this process, what was a flat ER membrane starts bulging out, as shown here. Whether this is mediated by these two proteins or there's something else going on remains to be, I mean, I'd like to challenge that this is driven almost entirely by certain specific lipids. But our understanding of lipids in this process is rudimentary at this stage. Now, the binding of cargos to 24 then at some stage leads to the recruitment of another dimer, which is made up of sec31 in yellow and sec31 in yellow and sec31 in this dumbbell shaped structure shown here. They bind. The binding of 1331 to 2324 triggers the, what it does is basically it potentiates or it stimulates the GTPase activity of sec23. So sec23 is a very weak GTPase, but this binding activates the activity, which can then hydrolyze GTP bound to SAR1 back to GDP. And that happens, this is paralleled to events that result in cutting of this part of the membrane, and you end up with a COP2 vesicle. Okay. Now, so remember, I've told you that over the last 3 decades, we've learned GDP to GTP starts the process, GTP hydrolysis to GDP terminates the event, and this results in production of a progeny, a COP2 vesicle. But Zanetti, Julia at Birkbeck College in London has recently uploaded a paper on bioRxiv, in which she shows that GTP hydrolysis is not required for vesicle production in yeast. So this really throws a big monkey wrench in the entire thinking that we've had to follow for the last 30 years. Now, in addition to that, these COP2 vesicles, you can visualize them in Saccharomyces cerevisiae, but they've not been visualized in metazoan cells. Why is that? There are 2 possibilities. It's a speciality of the Saccharomyces cerevisiae. Number 2, they exist, but they're a very minor population, or they're very, very transient. So those are issues that have come up repeatedly in discussions, but you know, so be it. But there's even a bigger problem. The problem has to do with the fact that you and I produce material that is far too big to fit into a conventional COP2 vesicle of 60 nanometer average diameter. Collagens of the type that are necessary for skin assembly, collagen type 7, for example, they can reach 300 nanometers in length, and they're hard, rigid structures. Chylomicrons are these lipid particles that you make in the lumen of the ER. They will bind to specific proteins like ApoB, for example, and they're exported from the cells. Their job is to go out and scavenge triglycerides and cholesterol, and then take it to respective tissues. Yeah. So, and then you have, of course, the mucins, which are very, very large molecules. They're not particularly large in the lumen of the ER, but when they get to the Golgi, they become absolutely humongous. Yeah? But it's the quantity that you're dealing with in this case. You secrete a liter per day by specialized cells called the goblet cells. So the question then becomes, how do you secrete this? And many other components that pose a similar problem. So so I'm giving you a kind of tutorial, a historical built up to where we are in this process. So in the early 2000s, when I was still at UC San Diego, we performed a genome-wide screen in Drosophila, and we aimed to identify new genes that would have a role in transport and Golgi organization, and we succeeded. And there were about 70 odd new genes that we identified, and we called them Tango for transport and Golgi organization. Now, there are 70 odd. It's unlikely that in my lifetime I'd be able to work with more than 3 or 4 or 5 to any level of satisfaction. And I'm going to present to you data on Tango 1. We have Tango 2, Tango 6, 11 that we're working on extensively. But Tango1 turns out to be very, very interesting protein. It is 1907 amino acids from its N terminus to its C terminus. It's a transmembrane protein. And we know that because it has a full transmembrane domain. It is also another transmembrane domain, but it doesn't span the entire membrane, but it is inserted into the inner leaflet. Okay. It has a proline-rich domain, 2 coil-coil domains. This part is mostly unstructured, but it doesn't form liquid condensates. Okay, so just to state it at the outset. It has a coil-coil domain. It has an SH3-like domain, but this is very different from your canonical SH3-like domain because it does not bind to proline-rich domains. Okay, so what is this protein then? First of all, this is the work of Kota Saito from my lab. So what he found, he was able to generate a very nice antibody to this protein, and he found that it's localizing to special domains in the endoplasmic reticulum. So this is the endoplasmic reticulum in red, and this protein localizes to 200 to 400 spots in a mammalian cell. Okay. Now, we've known over the years, for many years, that these are the domains where cargoes are collected from the ER before they can go out. Yeast does not have a bona fide ER exit site. So in yeast, you can exit from any place you want. But yeast does not, in fact, secrete a lot of proteins, whereas you and I are made up of cells that secrete bucket loads. So it sits at ER exit sites. It is a transmembrane protein, but what does it do? Well, Kota did an immunoprecipitation of this protein, and he found that the majority of the proteins that bind to Tango1 seem to be proteins of the ECM family, the extracellular matrix, and predominantly the collagens. He found many, many different collagens, but he focused almost entirely on collagen type 7 because there were 6 peptides corresponding to collagen type 7. It was the most abundant of the proteins that bound to Tango1 in his immunoprecipitation experiments. The next obvious thing to do was to deplete it. This is in 2007-8. And there was no CRISPR at that time, so we did RNAi, and you can deplete it. And you see there's about a 70 to 80% reduction. And when you test the effect of this reduction, there's no change in the organization of the ER, the Golgi, the small molecules that are secreted by cells were still being secreted, but collagen type 7 was present in very low amounts in the extracellular space. There was about an 80% reduction in the amount of collagen secreted. And what is not secreted is found accumulating inside the cells. And where does it accumulate? It accumulates in endoplasmic reticulum. So this is a patch of collagen that is prevented from exiting the ER when you have depleted Tango1. Yeah? But we were always very surprised to see a large collection of membranes very near the endoplasmic reticulum that seemed to be accumulating in cells that had been depleted of Tango1. We couldn't quite figure out what these membranes are, but they become very, very important. And I'll tell you why shortly. So this therefore gave us all we needed, that we have finally a protein that doesn't seem to care much about many of the cargoes that are exported by the cells, but it does control the export of collagen type 7s from the endoplasmic reticulum. And Kota was also able to show, he showed that the cytoplasmic part binds to Sec23. Remember, this is one of the code proteins. And he was able to do a very technically challenging experiment, but he was able to show that if you have a full-length Tango1, it binds collagen type 7. The luminal part of Tango1, which is in the ER, binds collagen type 7. The cytoplasmic part does not, which makes sense because collagen should never be in the cytoplasm. And if you have a full-length protein, but it lacks the SH3-like domain, then the binding is extremely low in quantity. So this gave us the idea that Tango1 is interacting with the collagens through its SH3-like domain. Okay, and it binds to Sec23 in the cytoplasm by its proline-rich domain. And others have now been able to co-crystallize Sec23 with the proline-rich domain of Tango1. So we then were left at that stage, and we published our story in 2009. In 2012, a group from Genentech in South San Francisco, well, they contacted me because they had been working on 50 genes that are highly connected to atherosclerosis. And they have tons of money. We don't. Well, we have plenty, but not as much as those guys. And in fact, Reinhard, who you must have heard earlier, a dear friend, he, well, he's speaking right now in another room. No, that's not true. You know, we've been traveling together, and you know, I said that no one would pay me the kind of money I would need, and vice versa, to listen to the same talk 3 times in a week. So we made a deal that I go to your talk once, you come to my talk once, and then you hang out. Anyway, so they're resting somewhere, and I was too in their talks. So we were trying to genetically delete this gene from the mouse system as well, but you know, we are not mouse geneticists, and we failed. We couldn't do it. But you know, these good people called me and they said they have deleted the mouse gene, and there's a phenotype. And I said, what is the phenotype? The phenotype turns out to be very peculiar. I don't know if you can see this. This is what a mouse pup looks like when it's born. This is what a mouse pup looks like when it does not have Tango1. It's basically devoid of mineralized bones. It doesn't really have any skin. In fact, many of the bones are completely missing. And it's short, it's half the size or so, and it dies at birth. That makes perfect sense. If you don't have collagens, you would have this particular fate. And they looked at not only collagen type 7, but also many of the other collagens. I can't remember the numbers, but many, not all 28, but many. So this was the proof we needed that what we had said based on our in vitro. I think cell culture system is in vitro. For those who think that this is real physiology, you know, be careful, yeah? But if you don't see a phenotype in a mouse, then you can always come up with an excuse, redundancy, et cetera, et cetera. If you do see a phenotype, that's good, right? So you can use it to your liking. So this is very good. This provided us with the impetus that we needed to move forward. So then something very peculiar happened. Then people started looking at Tango1 in patients, because if you have a problem with collagens, what would you expect? You would expect generic phenotypes of collagenopathy. The fetus would either be dead, as I showed you, or if there's a mild mutation, people or animals tend to be smaller, they have skin problems, they have skeleton problems. And so people started identifying mutants, and they contacted us, and we worked with 2 groups. And in one, the gene is deleted, and it's exactly the same thing as had been shown for the mouse system. In this case, the fetus is aborted 14 weeks after fertilization. It basically lacks bones. Yeah? And so understood that it replicates what had been shown in the mouse system. But then there are milder mutations that we've identified in collaboration with the others. What happens is you produce, there's a spliced variant, or there's a domain that is missing, et cetera, et cetera. And so what you do is you have an original molecule, but then you have another one, if it is a splice variant. And this one, shorter one, starts acting as a dominant negative. You secrete less collagens. You have generally people who, the kids who carry it, they continue, but they have mental disorders. They are very, very short. They're dwarfs, bent, the bones are deformed, and they have skin problems. Yeah? So good for our science, bad for people who bear these mutations. But it gave us the confidence that we are on the right path, and we should continue, because we would have finally an understanding of how cells deal with secreting these bulky molecules. So comes the big question, how? This is what I've shown you before. It's doable. It takes time, but it's doable. This is where you face the challenge. So how do you suppose Tango1 works? So I've told you that Tango1 binds to Sec23. It's binding through its proline-rich domain. Yeah, we found that Tango1 binds to collagen, or the SH3-like domain of Tango1 is required for binding to trimers of collagen. But a few years after our findings, a group in Oregon, Hans Backinger, showed that the binding is not direct. It is through a protein called HSP47. Now, HSP47 is required for export of many of the collagens, but not all. That means there are other adapters that remain to be seen. But it's important that this situation is getting a little bit more complicated. It's not so straightforward as Tango collects collagen and out you go. It doesn't work like that. Now, and I'm going to be very honest with you, and I think people who know me well enough to know that I think you have to be very, very fair in your presentations. So I'm going to show you, I'm going to present to you how we think the system works. There's a lot of data, a lot from our lab and many others, but there's also fiction. And when I say something, I will clarify it, okay? Because I think it's important. It gives you this fiction or hypothesis allows you to do the right kind of experiment. That doesn't mean it's written in cement yet. So this is fine. But the problem is, how do you then go from here to creating a structure which would be 400 nanometers to accumulate this molecule? How do you do that? In our field, all the understanding we have of a vesicle formation is that you collect membranes for creating vesicles from the departing station. So if it is clathrin-coated vesicle, you pinch membranes from the plasma membrane or the endosomes. For COP1, you collect almost all the membrane from the Golgi. For COP2, the small ones, regardless of their existence, they are supposed to collect membranes from the ER. Could it be that you do the same thing? All you have to do is basically pull a huge amount of membrane and you will get a big structure. But we could never demonstrate that. And there was another problem right at the outset that if this model is correct, then Tango should form a ring here in the plane of the membrane. So it should circle an area and then it will allow membranes to be pulled out in the cytoplasmic direction, much like the lingam of a Shiva, which is that structure that you see. That's what you want to create. And so we were struggling, and then comes one of yours, Ishir Raote. He is from this institute. He's now running his own lab at the Jacques Monod in Paris. So he comes to me one day in the morning, and he had drawn Tango 1 from the paper clips. And he says to me that here is the cytoplasmic part, here is the transmembrane, and this is the luminal part. And he says the coil-coil domains and the transmembrane domains bind, and they create a ring, as shown here, in the plane of the ER membrane. I said, come on, this would be a dream come true. He says it is true. And he could see by super resolution microscopy. I mean, it sounds very, very easy, but think about it. ER is spread out in a cell. We tend to visualize flat cells. It's spread out like that. You can look at your hands if you wear jewelry. If you were to look at it from the top, it's very hard to see a full ring. You'll see a small fragment, unless the finger is sticking out like so. And then you look at it from the top, and you might be able to see the whole circumference of a ring. And it's very hard to capture. But he's been able to do that. Oh, sorry. And you see there's a ring of a sort, there's a ring of a sort, there's a ring of a sort, there's a ring, there's a ring of a sort, there's a ring of a sort, there's a ring of a sort. These are many different images. And in the middle is the COP2 protein Sec23, as we had imagined the case to be. And the idea would be that this is in the center is where you would start creating a structure, which would be filled with collagens and going towards the Golgi. Now, how would you start this whole process? And this still remains unclear. But this is where we think we need to focus on now extensively. So the idea is that the collagens are synthesized as monomers. They assemble into trimers, but they assemble in a C to N terminus. The C terminus is always attached or in close proximity to the membrane, inner membrane of the inner leaflet of the ER. The idea would be that as it starts assembling into a trimer, it's pushing against the membrane, like so. And as it pushes, it creates a bulge. So you go from a flat membrane to a bulge as shown here, and this will continue. But this requires the Tango 1 to be stretched out to a length which is long enough to create this structure. So this is where a little bit of hard thinking comes into play, but the data for this is very hard to obtain. But we calculated that this requires Tango 1 to be a very, very large molecule if this were to happen the way you expect. The alternative is that you start the reaction as shown here, but the growth of this into a structure this big is through a different mechanism. So what did we find in collaboration with Jim Rothman at Yale and his postdoc Mao Han? we were able to do absolutely fanciest of the imaging that is possible. I think it's called 4-pi or pizza pi or something like that. I'm not a technologist, to be quite honest with you. But it took a very long time, so we were able to tag here, and then visualize it by their fancy imaging. And it turns out that this distance from here to here is about 50 nanometers based on the images. Okay? And this 50 nanometers means that only 25 nanometers of this would be sticking out into the cytoplasm. So there's no way this molecule stick out, or this part could stick out to create something that is 400 nanometers on its own. But about 20 or 25 nanometers is possible. So imagine then you have created, through this interaction between this part and pushing a structure which is now about 20 to 25 nanometers in height from the surface of the ER. This is not big enough, as you would appreciate. So how do you do it? So another postdoc in the lab who was working in collaboration with Ishir, his name is Patrick Earlman. He found that he went back and redid the immunoprecipitation of Tango-1 under more stringent conditions. And he found that, yes, he got all the stuff that Kotha had, but there was also always a protein that seems to have a function in the fusion of membranes. It's called SLY1. So fusion of membranes, as you know very well now, requires that a vesicle, when it has to fuse to a target membrane, it has proteins called V snares. You need a protein in the vesicle, and you need a set of, you just think of them as proteins present on two opposite membranes. They come together and they allow membranes to fuse, okay, VNT snares. And so we were able to demonstrate. So the point was, I never believed when Patrick Earlman told me that, why do you need fusion for fission? So you're trying to create a vesicle, but you're saying you need to fuse membranes. How does that make any sense? And he was able to demonstrate at least the involvement of these particular proteins. He showed that if you deplete SLY1, which is the protein he found in his immunoprecipitate, then there was a defect in secretion of collagen type 7. And what does not leave, in fact, remains arrested in the ER. So that made sense at that time, and it does to date, that fusion of membranes is required for creation of a compartment or a container to export collagens out of the ER. And so we were able to identify all the VNT snares, and all of that made perfect sense. That's a detail, therefore I'm not going through it. I think conceptually is what this is attractive, because it's completely new. So we proposed that some membranes in a cell are recruited to the site where you are creating a container for the export of collagens. Yeah? And these membranes need to fuse to this particular domain in order to create the structure of interest. So remember in my slide number 3 or 4, I told you that in the vicinity of the ER, there was always a large collection of membranes, and we couldn't figure out what those membranes are or where in our studies. These two good people, Patrick and Ishir, did the following experiment. They found that under normal condition, if red is the patch of collagen that needs to be exported from the ER, they found that this patch always was studied, or they were tightly opposed to the membranes that belong to a compartment called Urgic, which is a set of tubules and vesicles that live between ER and the Golgi. So this stands for ER Golgi Intermediate Compartment. This was identified by Hans-Peter Haury in the 80s. And no one could figure out what is the function of this particular compartment. And I'm going to tell you, because I think this compartment becomes very, very important. Think of it as a bank into which people put their money, and the bank uses that money to give it to those who need. So it's a source of membranes. Okay? For this source's membrane is used for making of autophagosomes. This source is used for trafficking membranes to the lysosome. This source is used for giving membranes to the ER, and this source is used for giving membranes to the Golgi. So it becomes a very important center, collection of membranes within a cell. Okay? Now, when you deplete Tango-1, collagen is arrested in the ER, and these membranes that are ordinarily tightly bound to the ER are now dissociated. This told us that Tango1 has the capacity to recruit ergic membranes. And we were able to map the recruitment. It's a very long experiment. We could express various parts of this molecule in the cytoplasm, but express it on mitochondria, which has no role in secretion per se. And we simply asked, can that domain recruit ergic membranes? And there's only one domain within this molecule of these 40 amino acids. If you express this part on the mitochondrial surface, there's a trick to it, but detailed, it could recruit ergic membranes. So this was therefore the proof that this domain has the capacity to recruit ergic to the site of synthesis of the transport intermediate. These membranes would fuse and you'd get a rapid growth in the formation of this transport intermediate. We call this tether for ergic at ER. For those of you who know Hindi, I don't, but you do, it's tir, that thing that you throw. That's what it is. So I have to, I try not to explain it to non-Hindi speaking people, but I thought you would have fun. So it is in fact now listed as that, as a specific domain for recruitment of material. So it turns out that in fact, this particular domain doesn't bind these membranes directly, but there is a set of components which are defined as tethers in our field. So these are very long coil-coil molecules. They function in binding, they sit on the membranes, and their job is to scavenge or search for membrane, and then they bring them to close proximity so that they can be used. So this tier domain within Tango1 immunoprecipitates 3 proteins, NBAS, RINT1, ZW10. These exist in a complex and they can stretch out very, very far. If you don't have the tier domain, that you don't bind it. So that doesn't mean the protein is dead, because if you don't remove a domain just proximal to the tier domain, it is still functional. Yeah? So this therefore gave us the following scheme to present to our colleagues, that Tango in green and red are the so-called tethers, can recruit ergic membranes in blue. We could visualize it. Yeah? We're trying to do this at the electron microscopy and cryomicroscopy level, but it's technically a bit challenging. So based on a lot of work, we proposed a model of this kind, that Tango1, this is not to scale, sorry, this is a bit dramatic to make the point. It doesn't work like that, but it's, in essence, this is how it functions. Tango1 molecules, by the way, Tango1 doesn't function on its own. It has cousins that help in the function of Tango1. So there's Tango1, which spans the ER membrane and has a domain in the lumen. Then there is a protein called CTH5, which is just like Tango1, but doesn't have any luminal part. And there's a protein called Tango short, which is just like Tango1, but again, it does not have a luminal domain. So they bind, they're in the membrane of the ER, but they haven't the ability to go into the lumen. Therefore, they cannot bind collagens. Only Tango in this trimer can bind collagens. But they're in the cytoplasm, they all serve the same function. So these molecules here, they bind to tethers, and this should be here. Unfortunately, this is in the annual reviews of biochemistry, so it's there to stay. There's nothing I can do. These molecules should be here and not embedded into the membrane. They bind to ergic membranes. The ergic membranes fuse by a snare-dependent process, and the fusion results in creation, a rapid creation of a structure which is big enough through which collagens are tunneled. So there is no need to create a vesicle. This is, in fact, the first cisterna of the Golgi, because this is almost a micron in dimension, and it is a composite of ER membranes. So part of it is derived from the ER, but most of it is derived from these ergic membranes, which come from COP1 dependent process. Membranes are being recruited here, and also by COP2 dependent process, which are being recruited here. Okay. So it's really a mixed cisterna. And now when this is cut, however that cutting happens, remember I just told you for 30 years we believed GTP hydrolysis was involved, and we learned recently, well, no, it isn't involved. So there is a mechanism that remains to be understood. And so we wait eagerly, and we are trying to address this ourselves, but for the time being, we don't really have an understanding. However, this material is either cut here or cut there. What you do is you end up generating a large cisterna, a large membrane, which is in fact the first cisterna of the Golgi. What happens is this large tubule or cisterna just keeps moving forward, and COP1 vesicles are bringing enzymes back for glycosylation of the material contained here, and they keep removing material that should not be going forward. So this structure keeps moving forward, and you're removing material that should not go forward. And it ultimately goes and fuses to the cell surface. So once you have created this structure at the level of the ER, there are no more tangos, and there is no more sorting per se in the forward direction. And this becomes what is generally known as cisternal progression model of secretion, meaning you don't remove material by COP1 vesicles to go from one cisterna to the other, but you're maturing cisterna, you're removing material so that only a pure form of a protein content can be released to the final destination. So this, therefore, completely changes the way George Palladi had envisioned secretory pathway, meaning the compartments are not fixed, they're moving, they're mixing, and what you do is you demix. So this then, you would agree with me, is a completely different way of thinking about how protein secretion or how proteins are secreted by the metazones. So if proteins are moving from here, you would say, well, you know, proteins can move along the membrane and proteins can move from here, or at least ions can easily move into this canal that you've created. How do you seal this structure? Yeah. So again, in collaboration with Jim and Fred Pentet in Paris, we did a very technically challenging experiment. This is done by Ishir again. So the experiment is as follows. He had shown that you need these transmembranes domains. They make a tight seal in the plane of the membrane. How do you test that? So he purifies this as one unit, shown here, and then just the transmembrane domain, shown here. These are then incorporated into a liposome, which contains fluorescinated lipids. They're green. You pull a tube, and then you bleach it. If the incorporation of these proteins had made a tight seal, so as you pull, these proteins will be attracted to the neck. If there's a tight seal, there'll be no recovery. But if there's no seal, you'll get recovery. Yeah. So you can test that. So here's the experiment: no protein, just the transmembrane domain. Here you have the transmembrane and the intermembrane domain. And you do photo bleaching and then look for recovery. We've only done up to, I think, 6 minutes. Yes, it's fast enough. Okay. So what you find is that if there is no protein, of course there's recovery. If there's only the transmembrane domain, it's a bit slow, but still almost complete recovery. But if there is the full set, the transmembrane plus the inner membrane, then there is hardly any recovery, or extremely slow. So this told us that this arrangement of Tango 1 as one full transmembrane and another one that inserts into the inner leaflet creates a very tight seal in the plane of the membrane. So the transmembrane proteins and lipids cannot move. We haven't solved the problem of luminal content, but we've solved the problem of the transmembrane content. So then you would ask a very simple question. So I'm just giving you kind of vignettes of how we are moving towards our total understanding of this process. So we wanted to model this, and Felix Campello, an ex-postdoc of mine, is a physicist by training, and he spent a lot of time trying to model how you would create this tunnel or a tube. So I'm not a physicist. I'm barely a biologist. And so he came up to me one day and he said, do this. You can do that. And he says, try to pinch. I remember I used to play this trick with my daughter. You can't pinch if the arms are really stretched. So the ER is highly stressed. How do you de-stress it so that you could pinch? Two ways. You loosen it from the connections. And it's very hard to do because it occupies 75% of the cell volume. The alternative is that you add membranes to a very specific site and you will loosen it, you will de-stress it. Now you can grow, you can pull. I can't, but imagine you could pull something out of your loosened membrane, sort of here, for example. And so you will keep doing it. You will pull, it'll get stretched again or stressed again. You will add more membrane, and you will do this in pulsatile motion, movement. And this is exactly what happens. Let me see if I can get it to work. So you will see green is tango, and red's the membrane that's growing inside. So you see it stalls, it's stressed. You add membranes, it grows, it stalls again, and it grows in this form. And when you look at these membranes by EM, I should have included that. I haven't a picture here. They always appear to be beads on a string. So these membranes are never smooth, long tubule for whatever reason. They always have that appearance, and that fits very well with what Felix Campello and Ishir and others had modeled in the system. Now, how do you push collagen into this ER membrane? So there's many ways to think about this. One would be the long arms of the Tango 1, which is 900 amino acid. You could envision it's doing this. So another way to look at this would be you go to Japan, you're waiting to board the trains, and these special people, what they do is they push you into the trains. They're doing that. Yeah. That wouldn't work because Tango is attached to the membrane. The alternative, it's a ratchet-like movement. That's what's happening. And that's exactly what happens. So we think, we again modeled this with Fred, Ishir, and my lab. We did that. So the idea would be that Tango 1 is bound to collagen. There'll be another one that will bind to somewhere else. And you just, it's a ratchet-like movement. But we need to test this experimentally. This is published. But what we are trying to do is to put a fluorescent tag here and another one here. If this is correct, then at some stage, they will show fret. Yeah, we can do it even more cleverly. So in a planaria called Nematostella, their Tango 1 has 3,000 amino acids, not 900. Why it has such a big luminal domain, I don't know. But one possibility is that when you have to regenerate, so you can chop the head off within a short period, it grows back. So the idea would be that it is required for massive export of collagens and other ECM proteins. So there it would become very easy. If you imagine you have 3,000 amino acids, then you put one tag here and one there, and if they ever come close, you should be able to do it by FRET or other means. So we're trying to do that, but the problem is expressing 3,000 amino acids into a mammalian gene, and we have to do it in the endogenous. We cannot overexpress it. It's not an easy task. We're getting there. So this takes us to this slide then, where this was the consensus for a long time, that you coat a part of the ER and you pinch and you get a COP2 vesicle. But I told you that we cannot see this, not just I. It's not that I have a poor vision, but others have tried and tried and tried and they don't see it. And this also does not explain how you would secrete bulky molecules like the collagens shown in this gray primer structure here. So we believe that most of the cells have a different mechanism for export of cargoes out of the ER, even including the small ones. And it is that COPs do not cover the entire structure, but they form a collar at the neck through the function of Tango 1. And what then happens is this area shown here recruits membranes from the ergic. They fuse and create an amorphous structure, which is now filled with collagens. And this is how the collagens are exported. So all you have to do is basically cut here, yeah? And this structure would then appear to be similar to the first cisterna of the Golgi, and this just keeps moving forward. So you don't really need vesicle-mediated transport in the forward direction. You only need it in the retrograde direction. Now, so then question arises, are all ER exit sites created equal? So there are about 300 to 400 exit sites. Are all of them firing at any given time and all used for export of the cargoes? The same cargoes, it's their division of labor. So Senasheri, who was a postdoc till about 3-4 months ago in the lab, she's a chemist. And we often thought that we should be able to address this issue by using a variety of tricks, and none of those worked. But we came up with this idea of using optogenetics. It's in principle very simple. So what you need to do is 2 molecules bearing 1 arm each of the 2 units that you need in the system. And when you shine blue light, they will bind. When you remove blue light, they separate. So what you do is you have Sec23 and you put a 1 arm of the 2 units that you need. And in Tango short, we haven't done it with Tango, but it's the same thing as I told you. You remove the proline-rich domain and you put the second arm of the 2 units that you need for optogenetically trapping of these 2 partners. They become trapped. When you shine blue light, they become bound to each other. And as long as you sit in the blue light, they remain bound. When you remove blue light, they separate. When you express this protein on its own, it's spread throughout the ER. Why? Because it needs proline-rich domain to bind to Sec23 for its retention in the ER. Yeah. So I can show you the experiment result. So pay attention here. So you have Tango 1 short without its proline-rich domain bound to M. cherry and eyelid. Well, a chimera, it's in magenta. It's spread throughout the ER. Sec23 is created as a chimera of this, and it is in green. Okay, now watch one, within seconds of shining the blue light, what happens? The Tango 1 short has basically been recruited from its diverse distribution into this punctate. Yeah, and they show almost perfect colocalization with Sec23. Now, if you were to remove the blue light, they go back. Yeah, so it's a very fast reaction. Now, what we can do is we can ask, are all the sites required for collagen exports? Are all the sites required for export of small molecules? Or is there a mixing? Or is there a segregation? So it's a complicated experiment, but I can give you just the final result of this experiment. So if we look at exogenous collagen type 7 expressed in these cells, or we look at endogenous collagen type 1, or we look at signal sequence GFP, this is a bulk marker, or a protein that is ordinarily secreted by these cells, these two molecules are exported from about 40% of the exit sites, whereas these people can go out from any exit site. What does this tell you? That there are certain sites that are specified for export of bulky molecules, whereas the small ones can choose whatever they want. And these bulky exit sites tend to be closer to each other, and they also tend to be closer to the Golgi. So the idea we have is that if this is the Golgi and this is the ER, then the exit sites that are predominantly used for export are here, okay, and not at the distal sites, because there you would probably have to make a very, very large tubule to come to this distance. So it's easy to walk 100 micrometers, but it is very difficult to walk 1 to 2 microns. So this is probably the advantage the cells have. Now, if you keep this light on for a very long time, then all of these sites start getting closer, 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 and they lump. And this is a microtubule and dynein-dependent process. Why they lump, or whether they become one large hole, we don't know at this stage. This is something that we are trying to do. In the last few minutes, I want to just sum up with some of the new stuff you had. Well, this is also new. These things should be in print very shortly. So the way I envision how the secretory pathway works and how the ER exit site function is as follows. So an ER exit site is not a site where you just create vesicles like so. An ER exit site is very specialized structure. It is somehow segregated from the rest of the ER, and it creates there is a large amorphous structure that emanates from this site, and it is filled with folded collagen, which is used for secretion. It also contains folded collagen that is degraded. So would you believe it, 15 to 20% of the collagens that you make are degraded. Why do the cells do it? We don't know. It's probably because the cells do not want to overload the extracellular matrix. You cannot have unused collagen in the extracellular space. That would be detrimental for building the right kind of ECM. Would you believe it that the same area is also used for degradation of unfolded collagens by the lysosomal pathway and by an independent pathway which requires autophagosomes. So these membranes then have the capacity to be brought to assemble from the ER exit site, and Tango 1 functions. But then there is a divergence. And how this divergence to different directions is obtained is currently not clear. But we think that this is simply that Tango and other molecules recruit other acceptors that provide some diversity in the final fate of the collagens and other molecules for their export from the ER, for their delivery, for either secretion, degradation, degradation, and degradation. Okay. So just 2 more slides. 3. So here's a problem. This has been a long-standing problem in the clinical aspect of human physiology, pathology. So if you have a cut, you want to seal it, and you seal it by secreting ECM. But if you keep secreting it, you get a hypertrophic scar. If you still don't stop, you end up with a keloid scar. And in a skin, of course, it's a blemish and you don't want it. It also turns fibrotic. But if you have it in internal tissues, the lung, liver, kidney, heart, you're dead. So 45% of deaths post-surgery are a result of hyper-ECM production, hypercollagen secretion. And there is no control per se at this stage. Depletion of TGF-beta was supposed to be the magic bullet, but it didn't work. Billions and billions of dollars were invested in that process. So we reasoned, being simple-minded, that why don't we just find a way to block collagen secretion? We could address this issue, and we did that. So Ishir is again involved in this process. So we created specific peptides which inhibit the interaction of Tango1 here, to its binding partner CTH5. They probably also inhibit the interaction of Tango1 to Tango1 and Tango1 to Tango short. We don't know that, but we know that they definitely inhibit this interaction. And these peptides are small. This is work done in collaboration with Thomas Krieg of the University of Cologne, Beata Ines, and Beata Eckes and Ines Neuendorf. They are the experts of skin biology that anyone knows of. And so we generated these peptides. They're small, 30 amino acids long. And what you do is you make a chimera with another short peptide, about 10 amino acids or 12, which has the capacity to take anything that's bound to it across the plasma membrane, but only as far as the cytoplasm, but not into the lumen of the ER. So you can use this trick to inhibit cytosolic functions, but not the nuclear functions. So we add these peptides to cells, and I'm just going to show you a few data. The first thing we do is we incubate the cells for 20 hours and we collect the supernatant. And we ask, is there a change in the secretome? So it turns out that many of the collagens are not being secreted. Yeah. And some of the laminins are also not being secreted. Here shown an example, laminin A4. Now, laminins look like a cross. They're even more difficult to understand how you would export them out of the ER. So this therefore provides an understanding of anything that's weird in shape and too bulky relies on Tango-like pathway for its export. So this was very good that we can use these peptides then to inhibit the function of Tango1. We can do this in, oh, by the way, yeah. So if you remove these peptides, the cells recover. And it takes about 48 to 50 hours, but they recover. So what was lost in terms of inhibition in secretion comes back. Okay. And we also measured whether there's any toxicity as a result of the peptides to the cells. There doesn't appear to be any. We haven't done it in a mouse model because mouse skin is not like human skin. And I'll tell you the right system to test this. So very good. And we can also do this. I'll just say, you don't need to look at the details. This was accepted 2 nights ago, by the way, this paper. So it'll be out soon. So it turns out these good friends of ours from Cologne, they have access to patients with scleroderma. So they can get samples of cells from scleroderma patients, and they measure collagens produced, and it's gobs and gobs of production of collagens. And you can make it even worse by adding TGF-beta, which potentiates the transcriptional regulation and production of collagen. So you start secreting bucket loads. Now, if you use our peptides, then you can inhibit the hypersecretion phenotype, and you inhibit hypersecretion phenotype of not only collagen or procollagen, but also fibrillin, fibronectin. So it appears that you're basically blocking the export of the ECM proteins by using this trick of ours. So this can also be then tested in a zebrafish model. And what you do is you take cold needle, frozen needle, and you make a hole in the skin of the zebrafish. When you make a hole, you start getting granulocytes and increase in the dermis, increase in the so-called basement membrane. And what these good colleagues of ours done, they basically injected at 5 spots around the damage done by the needle, peptides. And they look at recovery. And what they find is that you lower the amounts of collagen being produced and you reduce the migration of granulocytes. So you would prevent the hypercollagen secretion, and you would also prevent the migration of immune cells, which come and create an inflammatory response. So this works very well in this kind of system. So I think we are hopeful that we should be able to use this trick to address this issue of scarring, at least in the skin, if not internal compartment. So finally then, I write it like this: who would have thought? Because when we started this, there was a lot of challenge from the gurus of the field. So we have it finally, I think you would agree with me, because most do, that we have a solution to the trafficking of bulky molecules like the collagens. We have shown that the transport of collagens, and in fact, most of the cargoes out of the cells, is not by vesicles, small or big. It's direct tunnels, okay, between compartments. So there is mixing, but you do mix. Okay? Not all ER exit sites are created the same. There's a compartmentation within a compartment. How you do that, we are trying to address. And I've shown you that for the first time, we can target the secretory pathway, because people used to think secretory pathway is too generic to be targeted for any disease per se, but we can now do that by attacking one of the molecules, such as the Tango-1 molecules. Now, these are the people who have been involved in just the Tango-1 project. Fred Bard, French, he started the screen, he completed the screen. He used to be in Singapore as an investigator. He's a CNRS professor at Cancer Research, something else, Institute in Marseille, France. Kota was recruited first to Tokyo University as an assistant professor. He's recently been made full professor at this university in Japan. He basically gave us the Tango-1. Patrick Ullman was also a postdoc, so these are all postdocs. Patrick Ullman was a postdoc. He is now the head of research of Boehringer Ingelheim in the US. Very rich person, doesn't give me any money. Christina is at Oxford. Antonio, he's trying to make up his mind. He's an offer from two very big top universities in the US, and he's trying to figure out where he wants to go, but he doesn't want me to name the university. He'll be gone in short while. Ishir started his lab at this at Yakmano in last year sometime. Yeah. Felix is an independent investigator at another institute in Barcelona, and Sonashri, who did the optogenetics. I mean, when she wrote to me that she wanted to come to my lab, and I said, a chemist in my lab? What are you going to do? Make drugs? Well, she is kind of, yeah. So she had to go, she's gone to US, but she wants to come back to India. And if you're trying to recruit a person who is absolutely brilliant, nice, kind, generous, and ambitious, there's your catch. This is the lab. In addition to the people that I showed who've left the lab, many people, there's a postdoc missing because she's pregnant, and a student missing because he was on holidays, Alicio. And funding, we've been very lucky. We've had money from the ERCs and many other agencies, and that does it. Okay, this is it. Thank you very much.
Thank you, Vivek. Questions?
Thank you. My question is, what happened to the ergic membranes once they have been loaded on the on this tube? What happens to them? Okay, do they come back? And
no, they fuse. They fuse to the membrane. I think what ergic membranes do is there are two pools. There are distinct pools. A pool that is dedicated for growth of these kinds of structures, and another pool that can fuse almost anywhere to the ER. Yeah, we are trying to distinguish that. But most of them are used for this purpose.
And they generally originate from the ER.
So they generate, they seem, so when COP2 vesicles or when COP2 coats are inhibited in function, the ergic membranes disappear, meaning they rapidly fuse to the ER. Okay, there's a backward mechanism which is predominant. How that happens, we don't know, but the ergic membranes are receiving membranes from the Golgi. So one other possibility is, this is the structure, right? But as it is being built, you're also beginning to remove material that should not go forward. In fact, Jennifer published a paper recently, 2 years ago, where she showed that this tubule-like structure that comes out of the ER, this part is where COP2 coats are, this part is where COP1 coats are. So I think as it is being built, you're already beginning to extract membranes, which should, meaning you're recruiting and you're removing material which should not go forward. So the cells are very tightly controlling what should leave when you leave this structure from the ER. They're already engaged in recycling. So you're building, as you're consuming, you're also building ergic membranes.
Vivek, if you put a secretory tag on a protein, for example, and put it in any cell, typically it'll be secreted. If you put collagen in a cell where collagen is not normally expressed, will it be secreted?
No. So we are trying to make yeast produce and secrete collagen. These we need 200 genes for that because it's the folding.
But your cell, typical mammalian cell, exports other huge structures. So it should have all the enabling
if
environment.
we express it in human cells, yes, we can export collagen out of any cell.
Okay.
But I thought you meant if I put it in yeast, for example.
Okay.
So that doesn't work because it doesn't have tangos and many other proteins. But if I take collagen type 7, I put it in HeLa cells or even yeast, of course it'll go out. It'll go out because it has almost everything else. It's just that they're not used to seeing collagens, but they're used to seeing other molecules.
Okay.
Yeah. So one of the things that people have noticed that when a B cell becomes plasma cell, meaning it starts secreting antibodies, the levels of Tango 1 goes sky high. And if you look at old skin versus young skin has a lot of Tango 1. I think where Colin has done these kinds of experiments, and there's another group has done, and they're about to publish. So tell Colin to get cracking. So the young skin has a lot of Tango 1, but as it ages, the levels of Tango 1 drops. So you remember I told you that about 15 to 20% of collagen is never secreted but degraded. If you could siphon that amount and send it into the secretory pathway, there's a possibility of potential of perhaps recovering the collagen deficiency as noted in aging skin. So, rings.
So spot B. So what happens in stuff like COPD?
COPD has more to do with, there is fibrosis, but COPD has more to do with hyperproduction of mucus. So that's another.
So is this pathway hyperactive?
Yes, it is hyperactive, but we are working on the mucin secretions as well. And it turns out that the cells have a way to control mucus secretion, but all the genes that we've identified so far, Tango 1 is required for export of mucins out of the ER, so we can target, we're testing it, whether the same peptides can work. But all the other genes that we have identified, they sit at the cell surface. They control how many granules can fuse, the so-called condensing vacuole type. So Tango 1 has, these peptides have the potential of working not only in skin cells, for example, but we are trying to test them in airways. And that would be very useful.
Hey, Vivek, very nice. My question is regarding this ER exit sites. And you said about 40% of the exit sites might be involved in the collagen export. So if we imagine a scenario where multiple collagen molecules are being exported simultaneously, can you visualize this? Can you see these multiple tunnels being formed or?
So, it's all a matter of what a poor man can do. Yeah. So we have Sec23 labeled, we have Tango 1 labeled. We have a collagen. We've finally been able to engineer collagen with endogenous collagen with GFP. If you start overexpressing it, you create lots of problems. And we also want to visualize the membrane that contains them. And the kind of super resolution microscopy, and that has to be in living cells. The kind of microscopy we have to do for that is turning out to be very complicated. But this optogenetic trick is perfect for it. So when we trap, nothing leaves the ER. When we untrap, things go. Okay? But they go very fast. And we are trying to find ways to slow down so we can see a structure coming out, we could trap it. There we could do those kinds of experiments that you were saying. But till we do that, I think we'll just see a lot of material going out. But would that increase the number of ER exit sites? It likely does. Here is in that experiment I showed you with optogenetics. If you were counting, there are not 200 to 300 ER exit sites. They're close to almost they're more than 500 ER exit sites. They're numerous. So is it possible that when you overexpress Tango short, you have the capacity to make even more ER exit sites? We don't know any of that at this stage.
So in that case, do you think it will mess up with the Golgi organization itself?
It might, but for 40 minutes of blue light exposure, we see loss of ERGIC because it's gone all into the ER. There's no change in the structure of the Golgi. We are trapped. Yeah, so it's not that there is a fast recycling between ER and the ERGIC, but not between Golgi and the ER. We don't see any seminosidase looks more or less the same. We don't see it arrested in the ER. There's no recycling. There's no fragmentation of the Golgi either.
Yeah, I think you addressed a part of it because there's information flow from the ergic to the ER as well. But I was wondering, these long tubes, right? Do they also retract? Is the information always moving forward? Because I know ergic also looks at cargo, unfolded proteins, and then sends it back to the ER.
Well, I don't know if ergic, you can reach ergic for unfolded cargoes. I don't think you are allowed to leave if you're unfolded. You can't leave the ER. So I don't think this structure is doing this. And for us to do that, we really have to do it in living cells. I told you that we haven't been able to do that at this stage. In fixed cells, you'll never see that. But we are trying to do that experiment.
In live dynamics, ER ergic comes back to the ER and sends cargo back.
In what?
Ergic.
Yes.
Looks at a lot of unfolded proteins and sends it back to the ER for proper folding using heat shock proteins before it gets sent out again. This
Well,
is
you
from
know,
Hans-Peter Hauries.
I mean, the basic, I mean, unless something I've completely missed, when you are unfolded, you don't leave the ER. You don't go as far as the ergic. You know, this is what happens with CFTR, et cetera. You're stuck in the ER. You don't make it to the ergic. Ordinarily, what happens is proteins that do leave the ER, they're not necessarily unfolded. So some of the heat shock proteins under normal conditions would leave the ER. They'll go as far as the ergic and come back. But an unfolded protein does not leave the ER.
No, I mean to say in under stress situations, we can discuss this.
Well, under stress, so for example, if you shock the cells, heat shock proteins, you know, they come up. They're the first ones to come up. The levels increase. Some of those proteins go out of the ER. Some can be secreted. BIP is secreted. Okay? But most of them make it as far as the ergic and come back. They keep doing that. But proteins that are unfolded as a result of heat shock or damage, mutations, they cannot leave the ER. This is the whole process why you get unfolded protein response, is because they accumulate in the ER in an unfolded form and they mess up the milieu of the lumen of the ER, and then the cells respond by making even more proteins to take care of it. So there's a difference. So proteins can cycle, chaperones can cycle, but unfolded proteins do not leave the ER. You see,
No, I'm asking, we can talk about this later.
Yeah, sure, yeah. I'd be very interested in knowing if there are unfolded proteins that leak out of the ER.
I'm imagining the tango protein doing a real tango, because the cytoplasmic domain has got to leave this thing so that the ergic can fuse, and then the luminal domain has to push this thing.
So think of it like this. Here's a protein, right? So it's doing this. So I didn't get it. The transmembrane domain is very remarkable because if you make mutations in specific points, so the point of the cytoplasmic part is to do this. So if you imagine this, my bald head is the patch where you want to create the structure. It's doing this. Yeah, it's beating the head. If you make any mutations, it doesn't do this. It just does that. And that's a dead reaction. The luminal part is somehow conveying this message. It's a change in conformation. When it binds collagen, it does this. When it loses collagen, it does that.
Yeah. So now I'm just wondering, like, what's driving so much movement of this thing, both top and bottom? Is it just a consequence of the fusing of ergic and the binding?
The fusion of ergic comes later. It's part 2 in this reaction. This is allostery. When you bind, you change the conformation. So and people have been trying to figure this out since the days of Howard Shartman.
So what causes it? And it's got to be coordinated across all of those.
Yeah, I know.
Those that are.
I know. These are very, very hard questions, and I don't think in my lifetime I plan to see an understanding of that. But you know, we can now at least do the cytoplasmic part away from the ER exit site. We can now artificially express Tango1 cytoplasmic part on a scaffold at the mitochondria. And we can recruit the ergic and recruit the whole Golgi. And we can do some of the experiments that you've mentioned, but we cannot connect it to the luminal events. So we would have a situation where a protein would be, for example, in a permanently active form. So we can score some of the outcomes, but we cannot do what you're asking for. It's very hard. Yeah. But if there's anyone here who has a brilliant or even a good idea and you want to test it, come over.
Yeah, I guess in a similar lines, I was interested how this TEDR protein or this TEDR complex is regulated. I mean, is it like it's always bound to ERGIC, or it's like during the collagen transport, it is
So
also
this is what would
specifically-
happen. The Tango's luminal part will bind collagen. It will send information across the part in the lumen, across the transmembrane domain, and it will say to transmembrane domain, bind to Sec23. It will expose a domain, which will recruit tethers, which will then bring ERGIC membranes. They will fuse.
So recruitment of tether is like after all.
Probably,
during.
most likely, but we cannot distinguish those events at the level that we are looking at. Yeah.
Right.
Okay, I think we'll close here. We'll just thank Vivek for wonderful talk.
Thank you. Great, thank you.